Do you just want me to start, Tusi?
Yeah.
Yeah, okay, cool. Hello, everyone. Good morning or good afternoon, or I don't even know what to say because we are all in different time zones. So for some of us, it's going to be good evening.
Hello.
Hello. Hello, everyone. And I'm Anne Bent from SLING, the Singapore Lipidomics Incubator, and I'm one of the people behind this virtual podium. You see the organizing committees here represented on these pictures, but let's not fool ourselves. All of this only happens because the amazing Tusi got us all together for the third year in a row. And Tusi is just like this mastermind behind all of this and the driving force. So Tusi, thank you very much. I'd also like to thank everyone who submitted abstracts and also our sponsors who are sponsoring the the lightning talks. For today, the sponsors are the Steroid Biochemistry and Molecular Biology Journal and Westlake Omics. The virtual podium, I think we can go to the next slide to see. The virtual podium happens around the clock. You see all the different time zones and which are not good in which countries. That's why we decided this year for the first time to stream the recording in our evening, so it's more conducive for people in other time zones to listen to it. And on the next slide, we see the program. So as the same as last year and the year before, we have 3 days. We always have 2 keynote speakers and then 7 lightning talks. And for the first time, we also have tutorials. So I'm really excited about those. So the way we're going to do it is we start with the keynotes. We always have some discussion time. Also for each lightning talk, there is some discussion time. So Jurek and I, we are your chair people for today. And we hope for the lightning talks that you can keep it to 8 minutes so that we have 2 minutes for Q&A. And yeah, so we do all of these talks and then we have the award presentation and then the tutorials. So I think with this, we can start, right? With our first talk. Our first talk today is by Sarah Hancock, and Sarah Hancock is from Victor Chang Cardiac Research Institute. And Sarah really has a passion for using technology, mass spectrometry, to understand disease mechanisms. And she started with pancreatic cancer and then expanded to obesity and insulin resistance. And she's really pushing the boundaries, trying to go to like single cell. And we will see how far that has gotten so far. So today she's going to talk about FACS-assisted single-cell lipidomics of phosphatidylcholines and single myelins. Sarah, over to you.
I'll stop sharing.
One moment, I will share on mine. Hopefully everyone can see my slides.
Yeah, very nice.
All right, good to go. Thank you, Annie, for that kind introduction and for the invitation to come and present here at VPAP in the 3rd year that it's been running. And I'd also like to just start off by congratulating the organisers on putting together such a diverse range of speakers for the next 3 days. I'm really looking forward to seeing all the talks that are on offer. So as Annie mentioned, one of my interests is in mass spectrometry technology development. In particular, I'm trying to detect lipids from single cells. And so today I will be showing you some work I've been doing recently where I'm combining fluorescence-assisted cell sorting with high throughput nanoelectrospray to perform single cell lipidomics. Okay, so single cell technologies have really exploded in the last decade or so and have revealed a bunch of new information on fundamental biological processes such as cell differentiation, as well as some insight into disease mechanisms such as that associated with cancer. So in particular, where the technologies have mostly been developed is at the transcriptomics level of the central dogma of biology. So what these methods are doing is detecting RNA, and really this is enabled by the fact that you can amplify RNA for detection. However, data collected at the transcriptomic level really only describes the cell potential. And what I mean by that is that just because a gene is being transcribed doesn't mean it's being translated through to the protein level. And likewise, proteins can be post-translationally modified that changes their function or their activity. And so really where we might find more useful information is down at the metabolome level. So looking at things like sugars, nucleotides, lipids, and so forth. And this is because these things actually describe the cell phenotype. So what is going on in a cell at a given time. However, developing methods at this level is methodologically challenging because the metabolome comprises a wide variety of different molecules, different classes of molecule, different structures, and so forth. But they're also present at very, very low amounts. So this means that developing methods are perhaps not so easy since you can't simply amplify the signal of these metabolites. Nevertheless, there have been quite a few attempts, successful attempts now to develop methods that can detect metabolites from single cells. And I'm just going to talk through a couple of examples in the next couple of slides. So perhaps what is the most commonly used single cell mass spectrometry based method at the moment is that using matrix assisted laser desorption ionization, or MALDI. And so with MALDI, you have your cells of interest isolated on a slide, and then you are applying a matrix to those cells and firing a laser at it. So that then ablates the cell, and you can detect that using a mass spectrometer. So MALDI is particularly good for single cell analyses because of its resolution. It's roughly around the size of a mammalian cell, about 20 microns. However, the type of metabolite you are going to detect with MALDI is really dependent on the type of matrix that you apply. So just to highlight a key paper that's used this technique for single cell metabolomics, these guys from Europe recently have been able to use the technique to discriminate between two different cell lines cultured in the same culture plate, so co-cultured together, these HeLa and NIH3T3 cells. And using their method, or using some very sophisticated algorithms, they've been able to identify which particular cell type they're detecting from with about 90% accuracy. That's really, really quite outstanding. And then on the far right panel here, it's showing you the types of metabolite that are detected within these cells, with the ones on the right being those associated with the HeLa cells, the ones on the left, the NIH323s. And so what I really want you to notice here is that the majority of the things being detected are in fact lipid. Okay, the other method that's currently being used for single cell analysis of the metabolome are those using nanoelectrospray ionization techniques. And so really this is enabled by the fact that you can use a very fine nanoelectrospray capillary to directly pierce a cell and sample the cell contents. The group that has really pioneered this technique is this group from Japan, where they have coupled this nanoelectrospray ionization to microscopy, so video microscopy. So they term this video MS. And what this means is that when they're sampling from their cells, they can actually get spatial information as well. So, which particular cells have particular metabolite distribution present. And using this technique, they've been able to sample from several different key metabolites from cells, but also achieve a certain degree of subcellular localization by sampling either from the granules within the cell or from the cell body itself. However, I'm sure you can imagine with this technique, it's quite laborious to take an electrospray capillary and sample cells individually, so it doesn't really lend itself to being very high throughput. Some recent improvements to try and improve the throughput of nanoelectrospray ionization single cell analysis include the development of the single probe MS. So essentially, this technique uses these dual wall single probes that enable the continuous flow of solvent through the system to sample the cell and then through to the nanoelectrospray. And this is all hinged upon the stage system that allows these capillaries to raster across the cells and achieve a degree of mapping it to discrete cell populations, so a degree of imaging, I guess. So just to summarize the current state of the literature on single cell metabolomics, this table I put together a little while ago now, so it probably needs a bit of updating. However, really, you've got surface-based techniques and fusion-based techniques, and I've just highlighted a couple of key papers from each for you. MALDI mass spectrometry is by far the most popular. The challenge of using this technique, however, is that the type of metabolite you're going to detect is really dependent on the matrix that you use. Whereas for nanoelectrospray ionization-based techniques, you're faced with challenges such as the low throughputness or not being able to generate a stable enough spray for a long enough period of time to acquire MSMS data that is critical for metabolite identification. So taking all of that into account, when I was trying to put together my method or come up with the method that I wanted to use, I really wanted to overcome some of these limitations, but also try and introduce some new ways that we can characterize or map back to cell heterogeneity, the cell phenotypes. Okay, so this is the method I ended up coming up with. I've coupled the fluorescent assisted cell sorting with nanoelectrospray ionization. For this work, I've been growing cells in culture. For the method development, I use CTC12s, which are a myoblast cell line, and HepG2s, which are hepatocarcinoma cells. I then perform the FACS to enable the single cell sorting, and I'm just sorting those cells directly into a well that's already got a little bit of methanol and a little bit of antioxidant in it, as well as some internal standards. So it's really quite low on the cell handling side of things. That gets sorted directly into a 96-well plate, which I can then take over to the source of my instrument, which is an Advian triversa nanomate. So this is a chip-based nanoelectrospray source. It has a robotic arm that can sample directly from the 96-well plate, and then it infuses it through this electrospray chip that has these very, very fine narrow bore holes in it, which mean that you can generate very, very low flow rates, so picoliters per minute or less, meaning that you can generate a really long spray for a really long time. So I'm generally running up to 30 minutes of spray acquisition time, but you could go up to 60 if you wanted to. On the back end of this system, I have a SciEx QTrap 5500. So this is a hybrid triple quadrupole linear ion trap system. And I'm performing shotgun lipidomics using a technique that relies on complementary precursor ion scanning. So what do I mean by this? For those of you who do a lot of mass spectrometry work, you know that triple quadrupoles can be operated in a bunch of different modes, but typically a lot of people are using them for targeted metabolomics using multiple reaction monitoring, where you're fixing the first quadrupole on a specific mass on a precursor mass, and then fixing the third quadrupole on a product ion mass. However, what I'm doing is I'm actually utilizing the precursor ion and or neutral loss scan modes of this instrument, where I'm setting Q1 to scan for things that produce a fixed product ion mass. And this really allows me to maintain that sensitivity of a triple quadrupole, but also have some semi-targeted capabilities. So when I do this, for example, so I fix that third quadrupole onto a precursor ion mass of 184, and I restrict the mass range to this area, I can detect all lipids that contain a phosphocholine head group. And so really what I'm detecting here are things like phosphatidylcholines and sphingomyelins. Okay, so that's how the method works. On the back end of this is data analysis. I'm identifying my lipids using SciEx Lipid View software, and then performing some post-processing in R to do the background normalization, quantitation, and all the statistics. So I actually presented this data a few years back at ISLS, the Singapore Lipid Symposium. And one of the questions I got asked was, does FACS have any impact on lipid profile? And I thought that's a very good question. I should probably figure that out. So we know that FACS can impact on the polar metabolome, but little is known about the lipid. So what I did is I prepared some extracts from my C2C12 cells the traditional way, these bulk cell extracts where I'm growing them in a 6-well plate to about 1 million cells per mil, and then performing an MTB-based extraction to get my lipid. I then take that extract, so I have about 1 million cells in a mil at the end of that, and then I dilute it 500 times to get it within the range to perform shotgun lipidomics. So the range where I'm not saturating the detector or causing any problems for the ion optics. When I do that, I get the following scan, precursor 184 scan, looking at which shows me all of the different phosphatidylcholines and sphingomyelins that are present in my cells. I then did the same analyses on 50 cells sorted by FACS, so 50 cells per well this time. I didn't need to perform any dilution. That actually got it down into the reasonable detection range, and produced the mass spectrum shown here, precursor 184 again. So as you can see, just eyeballing between the two, there are very little apparent differences present between the intensity of the phosphatidylcholine and sphingomyelins that we can detect from these cells using the two different techniques. Just to put your mind at ease though, I actually analysed n equals 3 of both, put the statistics together on this slide here in this bar plot, just to confirm for you that in fact, FACS does have very little effect on the lipid profile. Okay, that's all well and good. I've shown that my method can, the FACS anyway, at least doesn't impact on the lipidome of these cells. Now let's use it to detect some lipids from single cells. So just to start off with here, I've got my C2C12s again. This is that 50 FACS sorted cell population I showed you before. I've now added some internal standards and I'm showing you the traces from 3 different replicates of these 50 sorted cells. We do the same thing with single cells. On this particular plot, what I'm showing you is the spectrum from 6 different single cells. Again, we've got some internal standards added in, 1.83 femtomolar for anybody who would like to sort of estimate what range we're looking at here. And as you can see, the types of phosphatidylcholine and sphingomyelin I'm detecting between the two are very, very similar, albeit the signal to noise is a little bit poorer, but still within tolerable range. You know, things are mostly above a signal to noise range of 4. Sorry, signal to noise ratio of 4. So that's all well and good, but you could still look at these data and think, well, perhaps that is just noise or background from the instrument. So to fully establish that the method is detecting lipids from single cells, I next performed an experiment where I incubated my C2C12 cells overnight in the presence of docosahexaenoic acid, or DHA. So DHA is not a lipid that mammalian cells can make easily, and there's really no way the cells can make it without supplying it in the media to them. Sure enough, when I incubate my cells overnight in the presence of DHA, we get this shift towards phosphatidylcholines that contain 6 or more carbon-carbon double bonds. So this just indicates that the lipid exposure is working. And again, looking at our single cells, not surprisingly, we can see that there is this shift as well in phosphatidylcholines containing 6 or more carbon-carbon double bonds. So this really demonstrates that the lipids that my single cell method are detecting are actually lipids. It's not just noise. And also that, you know, the method has the power to discriminate between some biological differences between cells treated, you know, without exogenous fatty acids. Just to summarize these data for you, I've been able to detect more than 50 discrete phosphatidylcholine and sphingomyelin species at the sum compositional method level using this method. I'm just showing you here the data again for the C2C12s. Top panel are my control cells, bottom panel are my cells treated with DHA, and we've got all the different lipid species detected across the x-axis. And then I've got the intensity plotted here, quantified lipid amount. So the darker red, the more lipid is present. I also performed the same thing for my hepatocarcinoma cells. Sorry, one step backwards. Just to confirm for you again, the DHA treatment is working. We can see that there is enrichment of phosphatidylcholines containing 6 or more carbon-carbon double bonds. I also performed the same analysis in a second cell line, so my HepG2 hepatocarcinoma cells. Took all that data, plugged it into TS&E, which is what I'm showing you here, T-stochastic neighbor embedding plot. And what you can see is that each of my cell lines and treatment groups cluster quite nicely, showing that the method can discriminate between both different cell lineages as well as different cell treatment types. However, there is some heterogeneity present in the data. You can see, for example, in the C2C12 control cells, we've got a couple of things that are clustering from the different cell lines, so both the HepG2 control cells, as well as the different treatment groups, so the C2C12 DHA treated. So this indicates that there is, in fact, heterogeneity present within my cell populations and that we can detect that using this method. Okay, that's all well and good. Now let's take that method and apply it to something where the changes happening to lipids are a little bit more subtle. Just try and push this method to its limit, so to speak. And so looking at different applications we could use, my group, as Annie mentioned in the intro, is very interested in cancer cell metabolism. And I decided to test this using prostate cells, simply because we know that the types of lipids present in prostate cancer are highly correlated with their malignancy or their ability to metastasize. but also the different established prostate cell lines, there is quite discrete differences in the abundance of different phosphatidylcholine present. So just taking a look at one of those phosphatidylcholine as an example, PC34-1, we can see, for example, in the PC3 cell line that PC34-1 is present at about 25% of the total phosphatidylcholine, whereas in a cell line like LNCAP, it's more like 45 to 50%. So I was able to get a hold of 3 different prostate cancer cell lines, DU145, LNCAPs, PC3, as well as 1 additional non-tumorigenic prostate cell line. And I performed the same 50-cell FACS sorting and just took a look at the relative abundance of the different phosphatidylcholines just to check that my values were in line with the literature. Sure enough, our PC3 cells, phosphatidylcholine 34-1, about 25%, whereas for the LNCAPs, it's more like 40 to 50%. So that's great, aligns with the previous literature. I then performed a single cell analysis across the 4 different cell lines from 18 single cells, which is what I'm showing you in this panel B here. And so this is expressed as relative intensity simply because when I quantified these data, you couldn't see the LNCAPs, for example. It's just really, really low levels of lipid that are present within these cells. Took all that data again, plugged it into my TSNE plot and got the following as shown in panel C down the bottom left here. So compared with the previous TSNE plot I showed you for my prior experiment, you can see the clustering is perhaps not so neat. So there's a little bit more heterogeneity present within these cell lines, but there's still reasonably good clustering. We can still separate them out into 3 broad groups, PC3s in green, an overlap group of DU145s and PNT1s in the middle, and the LNCAPs on the far right in blue. And so this actually corresponds really well with the ability of these cells to metastasize, or I guess which ones have more active metabolic states. The PC3 cells, for example, are quite active metabolically, whereas the LNCAPs are more quiescent. So it's really nice to see that some more phenotypic data is coming through when I'm applying these analyses. Okay, so just to wrap up what I've shown you here today. So I've developed this method for performing single cell lipidomics. It combines fluorescence-assisted cell sorting with high throughput chip-based nanoelectrospray ionization and shotgun lipidomics. We can use this method to detect biological heterogeneity that's present between cells of different lineages, as well as cells that have been supplied exogenous fatty acids. And we can also use the method to study heterogeneity present in some situations where the lipid differences are a little bit more subtle, so prostate cancer, across different prostate cancer cell lines. Where I'm going to next with this, so of course I want to expand the coverage. I'm actually on parental leave at the moment, so when I get back in the lab next year, I plan on trying to develop this method further to more lipid classes as well as polar metabolites. I'd also like to start using this method to interrogate, so using the FACS capability, interrogate what is the cell phenotype in terms of cell markers, correlate that to the lipidomics to get even more enhanced data on cell heterogeneity and phenotype. And because we're a lab that's really interested in cancer, I want to take this to the clinic eventually. So where I can see this being particularly useful is for analysing liquid biopsy of circulating tumour cells from different cancer types where lipids are a factor. Last slide, I want to thank everybody that's been involved in the project. My PI, Nigel Turner. We were at UNSW Sydney when this work began, but we're now at the Victor Chang Cardiac Research Institute. RA Eileen Ding, who kept this work going while I was on my first parental leave a couple of years ago. We've got Emma Beeves from the Mark Wainwright, who does the FACS for me. And of course, Todd Mitchell down at the University of Wollongong, who continues to let me go down and use his instruments. Of course, got to thank the funding bodies for this work. And I'm happy to take questions.
Thank you, Sarah. That was beautiful. It's also really amazing how you get all this work done and squeezing out 2 babies at the same time. So congratulations on that too.
Thank you.
I don't know, it's fascinating. So did all of this work primarily on the triple quads?
Yeah, all done on a triple quad.
Yeah. Yeah, so cool. I have another question. Let's take a question from the chat. I have a question regarding the TSNE plots. Are they same as the PCA or PCOA?
Well, okay, so I'm not a statistician, full disclosure, before I answer that one. So I'm really, standing on the shoulders of giants when I'm looking through and running statistical analyses. As I understand it, there's similar mathematics underlying both the TS&E and PCA. However, TS&E really tries to reduce the distance between the variables that you're plotting, whereas PCA tries to expand on that. PCA is really looking for that variance where it's present in its different components. TS&E is trying to condense that down. So I think, you're running these plots, you do tend to get very similar plots between TS&E and PCA. Of course, TS&E is a little bit more randomly generated. So each time you generate a new TS&E plot, the plot will look a little bit differently. Hopefully that answers your question. But again, I just want to make it very clear, I'm not a statistician. I spent a lot of time reading on statistical methods, but don't feel that I am an expert on this by any means.
Oh, you're on mute and
Sorry, thank you. Bishwa, I said you want to ask your question.
yes, I have a couple of questions. Sarah, great, fantastic work on single cell lipidomics, where there are very little studies. But the question is, lipids are very much structural, many of these, or majority of them, and there are functional lipids too. So facts, I have a hunch somewhere, or I believe that it may not be changing the structural lipids, but they could be doing a lot with the functional lipids, unless you have that done in a functional study where you have that annotation done and categorization in terms of a disease study or a functional study. I think it's very early on to infer that the lipid changes are not impacting because of the FACS process. You have any comments on that?
Yeah, sure. I think it's a good point. Lipids can have multiple functions within a cell. The ones that I'm looking at, phosphatidylcholine, sphingomyelin, very much structural lipids within the cell. They do have some signaling capability though. PLA2, for example, can cleave fatty acids from the SN2 position of phosphatidylcholine. Things like arachidonic acid and other polyunsaturated fatty acids can participate in signaling reactions. And we're not seeing a huge change in those, which leads me to believe that the FACS isn't affecting those specific pathways. But you're right, there's other lipid classes that are signaling-based. ceramides, for example, come to mind. Once I can get the method able to detect ceramides, perhaps we can start to interrogate that a little bit more fully. But yes, thank you for pointing that out. I think that's very good to clarify that.
Absolutely. Thank you so much. But the second quick follow-up question is, what is your yield in terms of these cellular manipulations? What kind of millions of amounts of cells you start, and what are the losses because of cellular frailty, using FACS as a method? Do you have losses in terms of cells? The yield is too less, is manageable? Yeah. Thank you.
I haven't tested that specifically. Every single well that I've injected from, I get some sort of signal, whether that's lipid or background. I do background normalization, right? So it shouldn't be background. You could probably see in some of the heat maps that I showed that there were lines of cells where perhaps the signal wasn't so good. So maybe those are cells that have, the FACS hasn't worked properly, or they've died, or something's happened to them during the FACS process. But they're very easy to discriminate, right? So I would have to think of a way to test that. I'd be open to any suggestions you might have offline, Biswa, how I might do that, because I think it's very important to ensure that the method is robust, as you say.
Cool. We could take one last question from the chat, but in the interest of time, after that we should move to the first lightning talk. Latoya is asking, are you running positive-negative switching?
Great question. All these data were acquired in positive ion mode. That's why I'm only able to detect the some compositional phospholipids, so phospholipids with their total number of carbons and double bonds. To get the molecular pairings, so the fatty acid pairing on those, I'd have to run in negative ion mode, which is a little bit less sensitive on this instrument than positive ion. So I haven't been able to get it working for negative ions just yet. Hopefully one of these days I'll get some nice grant and I can upgrade to a 6500 plus, or perhaps even the Xenotoph if I'm very lucky, and maybe push that out a little bit further. But great question. Thanks for asking it.
Cool, thanks. I think we move on. Sarah, if you can stay in the line, maybe you can continue to answer the questions in the chat.
Yep, I'll be able to stay online.
Cool.
Thank you, Annie. Thank you, everyone.
Thank you very much. So with this, we move on to the lightning talks, and our first speaker is do I pronounce your name correctly? Xiao Liang.
It's Xiao Liang. Yeah,
Xiao
exactly.
Liang. Thank you.
Fantastic.
From University of Westlake. And that's the next slide. Yeah, and he's going to talk about proteomics investigation of diverse serological patterns in COVID-19. Take it away.
Yeah.
You've got 8 minutes and then 2 minutes for discussion.
Great. Thank you, Anne, for the introduction. Wow, let me share my screen. Can you see my screen now?
I think it's starting. We don't. Yes, now we see it. Yeah.
Oh, great. Wow, it's so great to participate in this year's VPAP. Thank you. And it's great to meet so many familiar faces. Especially thank you, Susie, for inviting me. Okay, so yeah, so I will present my research in the form of a poster. I think my title is blocked. Okay. So I will introduce our most recent COVID-19 research that utilized proteomics to investigate the diverse serological patterns. Okay, so as known to all, our body relies heavily on the secretion of serum antibodies to defend against the SARS-CoV-2 attacks. And it has often been seen that patients with unexpected serological pattern, for example, the negative antibody expression or exceptionally high expression of IgM or IgG that are observed, which we think suggests the highly diverse host responses when having COVID-19. However, this type of molecular mechanisms behind them remain to be assessed. So in this study, we procured a clinical cohort of 144 COVID-19 patients and assessed firstly their 2-year antibody expression dynamics. As seen here, the x-axis, I don't know if you can see my mouse, but yeah, the x-axis denotes the weeks since the onset, and the y-axis denotes the IgM and IgG expression respectively. And here, the dashed line denotes the cutoff for positivity. Based on their expression characteristics, we classify the patients into 4 groups. So as shown here, the green line remained IgM and IgG negative throughout the disease stage, and we named it as the G-N- group. The yellow line remained IgM negative, whereas IgG seroconversion, and we named it as the G+N- group. The other 2 groups are all IgM and IgG positive. However, the purple line compared to the blue line are more tenfold in expression when reaching the IgM plateau. Therefore, we named them as the G+N+ group and the G+N++ group, respectively. As for their 1-year revisit, as shown here as R1, all of the expressions, regardless of IgM or IgG, experienced a significant decrease. Over 80% of the patients received vaccination between R1 and R2 2-year revisits, and still most of the patients experienced a significant continuous decrease of IgM at 2-year revisits. As for IgG, we classified the patients based on their vaccination status into the VAC and non-VAC group and found a significant boosting effect of the IgG titers after vaccination. We dug further by assessing the clinical information and found that all of the G-N- patients are non-severe cases. On the contrary, over 1/3 of the patients with positive IgG and IgM are severe cases. As for their admission expression of the clinical indicators, we found characteristic patterns such as the inflammatory factors such as interleukin-6, 10, interferon gamma, and acute phase proteins such as ACA, CRP, and coagulation factors, fibrinogen, that are all positively correlated with antibody expressions in general.
On the contrary, the lymphocyte counts and the nutritional factors, albumin and pre-albumin, are negatively correlated with the antibody titers. We dug into more details and found that several CD molecules, such as the tissue surface markers, the CD3, 4, and 8, that are all lowest expressed in the G+, M++ group, whereas the highest expressed in the G- M- group. As for the BCL marker, the CD19, it follows the similar trends. We also found some interesting diversities in the lipid-related molecules, such as triglyceride, total cholesterol, LDL, and HDL. Okay, to go deeper into the molecular insights, we collected 111 serum samples from 16 characteristic COVID-19 patients, as shown here, and identified in total 1,600 proteins using the TMT Pro labeling technology. As for their proteomics correlation, we found a high correlation, high intragroup correlation in the G- M- and the G+ M- group, suggesting that their serum host responses are less alternated during the disease. As for their differentially expressed proteins, the depths, over 60% of the depths come from the first 2 weeks, which can be utilized to easily stratify between the G-, M- and the G+ M++ group. Some of the characteristic depths can be utilized to construct an interaction network using string. And the most enriched functions during the first 2 weeks are the complement system and the prothrombin activation, which includes some of the characteristic proteins such as the complement 1 components, C1QB and C1QC, as well as the IgD antibody. During the late stage of the disease, at week 7 to 10, the most enriched functions include nucleoside extravasation, which includes VN1, HDL regulator, and FcGR3A, a regulator for the cytotoxic effector cells. We also assessed the correlation between the depth and the antibody and found one protein, CD163, which is a marker for the macrophage that were continuously upregulated in the G+ M++ group till after week 4, suggesting a more activated macrophage polarization in these patients during the seroconversion stage. An example of our depths with the lipid-related molecule is shown here, suggesting plausible lipid involvements in the immunological activities during COVID-19. By combining the above evidence together, we proposed a putative working model for the antibody diversity. According to our data, the complement cascade was initiated upon disease onset, which activates a list of inflammatory factors. This process activates macrophage differentiation, which further modulates local inflammation, leading to the activation of such as neutrophils, T cells, and eventually the B cell maturization into plasma cells. This whole process was not well activated in the G- M- patients, probably due to a prior cellular immune responses that may efficiently confront the invasion of SARS-CoV-2. On the other hand, for the G+ M++ patients, the cellular immunity may not be enough. Therefore, a high expression of the inflammatory factors and rapidly ascending IgM titers may be a complementary process to defend against viral attacks. That's all for my research. I'm open for any questions. Thank you.
Thank you. Pretty cool. So, so among those longitudinal samples, were there any long COVID people?
Wow, that's actually a very good question. We had a non-COVID paper, specifically a separate non-COVID paper, but in this paper we didn't assess the long-COVID because for 2 reasons. Firstly, the long-COVID include over 200 clinical symptoms, which makes it very chaotic. And secondly, the evidence we can find from the about non-COVID from the serum is quite limited due to the relatively low protein identification.
Yeah, makes sense. Oh, I see already 2 questions for you in the chat. What were the confounders, age, sex, diet, lifestyle, accounted for the protein abundance analysis across the 111 serum proteomes?
Oh, I'm sorry, but I had to check the. Okay, so Mr. Misra asked a question about the confounders for the protein abundance. Thank you for your question. And yeah, this is one of the main limitation of our study due to the too limited sample amounts. Yeah. And there are also 2 reasons. Firstly, it's kind of difficult to conduct COVID-19 omics research due to the restrictions of sampling collection, and secondly, this clinical cohort is kind of limited, which contains only 144 COVID-19 patients, where we collected 111 serum samples for the omics profiling.
Cool. We could take one more question by Yurik, and then we need to move on to the next speaker, please. Is there, see Yurik's question, any pharmacological treatment differences among the IgG and the IgM groups?
Oh, yes. Actually, as we can see here, because over 1/3 of the patients who are seropositive with severe cases, they receive more severe medical treatments of such as immunoglobulin and other antiviral drugs. Yeah, and that's why we specifically checked their on-admission expression of clinical indicators just to eliminate the effects of drug uses.
Okay, cool. Thank you again. If you see other questions for you popping up in the chat, maybe you can continue to answer questions if more pop up.
Great, thank you so much.
Thank you again for your nice talk. With this, I'd like to invite our next lightning talk speaker. Miss, I'm sorry, I hope I pronounced your name correctly. Gizal Siddiqui from the Monash Institute of Pharmaceutical Sciences at the Monash University.
Yes, thank you, Anne. Pretty cool.
Pronounce your family name.
So I'll just share my screen.
Yeah, go ahead.
Hopefully you can see that okay.
Yeah, yes, now it's perfect.
Okay, brilliant.
Right, cool.
So thank you for the opportunity to present today. So today I'll talk about some of the omics techniques that we've established in our lab in order to look at Plasmodium falciparum and its ability to be affected by these peroxide antimalarials. So my focus is mostly around omics, but also my main research interest is malaria, which the parasite that causes malaria is known as Plasmodium falciparum. So the current antimalarials that we have in the pipeline is these peroxide antimalarials, with the artemisinin being the frontline gold standard antimalarial against the parasite. These artemisinin derivatives possess this 1,2,4 triaxon core with a peroxide bond, which is absolutely essential for its activity. So if you get rid of this peroxide bond, these compounds are no longer active within the parasite. Now, with these peroxide bonds, it's highly reactive, and in the presence of heme and iron inside the red blood cell stage of infection, it gets activated, it forms this free radical, and this free radical then goes on to alkylate nearby proteins and small molecules, resulting in the death of the parasite. Currently, the antimalarial that's in the pipeline is this dihydroartemisinin. But problem with this dihydroartemisinin is that it has quite a short half-life in human, less than an hour. And now this peroxide bond was used for the synthesis of this synthetic peroxide antimalarials, and we collectively know them as the ozonide. So there's the first generation, which is OZ277, and you can see it has these chain addition, and it had a significantly longer half-life, and it's approved for use in India in combination with paraquat. The second generation, OZ439, is currently going phase 2B clinical trials, and it's combination with paraquat and has an elimination half-life of actually 46 to 62 hours. So this is significantly longer than dihydroartemisinin. So in our lab, we were interested to see what are the peroxide antimalarials protein target. So we decided to do this using, 1 minute. So we decided to do this using this chemical proteomics approach and compare temporal protein alkylation profile of our clickable peroxide probes. So what I would like to show you, so these numbers here is the IC50 or the killing value that we get within infected cells. So they're in nanomolar range, and we used a probe that had the alkyne handle, and I hope you can appreciate that the value in the cells and for IC50 is quite similar to the parent compound. And again, so this is for the simple ozonide, so which we'll call as the first generation. And then we also had an alkyne probe for the second generation of the ozonides. We had some controls, and one of the controls that we used was this Carba OZ277. And so it did not have this endoperoxide bridge, which I mentioned is essential for its activity. Therefore, the IC50 for this was greater than 10 micromolar, while these are all in nanomolar range. So our approach also used a number of other controls, and we used a data-independent mass spectrometry-based approach for identification and quantification of protein targets of these antimalarials. So as I mentioned, we use these probes, which I will call them the 3 probes. So there's the artemisinin, the second generation, and the first generation of ozonides. And we took samples within parasite over this many hours. We had to do this experiment in live parasite because, as I mentioned, the drug needs to be activated, and this activation is mediated by heme and iron. So within the red blood cell stage, the parasite actively takes up a host hemoglobin and digests it for its de novo protein synthesis. And during this process, it releases heme and iron as a byproduct. Now, this heme and iron is actually what causes activation of this compound. So we then use a click chemistry-based approach to affinity purify and do on-beat digestion of the protein target. And again, did data-independent acquisition, mass spectrometry, and the analysis was done using Spectronaut and a reference spectral library, which I had generated in the lab. So some of the other controls that we used, so we did competitive inhibition with the simple ozonide. So we added a different ratio of just a simple compound on its own to look for competitive inhibition of the protein target. We also blocked this hemoglobin digestion, which is essential for activation of the compound using this enzyme known as E64D. So what we found was that these antimalarials actually have 435 parasite protein targets. So that's quite a large number, but that's anticipated because these compounds are highly promiscuous and they have a number of targets. And hope you can see from this heat map is that the proteins that were enriched in these probes was significantly less in our appropriate controls. And so when we had more of the simple ozonide, as you can see from here, the enrichment was less. And also if you inhibited hemoglobin digestion, the enrichment was less. Suggestive is that of the fact that these protein targets are actual alkylated targets of our peroxide antimalarials. We then went on to do Go analysis of the cellular compartments that are being enriched. And so what we saw was the food vacuole was enriched. Now, as I mentioned, hemoglobin digestion and that release of heme is important for the activation of the compound that take place in the specialized organelle of the parasite known as the food vacuole. So the fact that the cellular compartment and the food vacuole is being enriched is not surprising to us. What we found surprising was that actually the cell redox homeostasis was also being enriched. So what I'm listing here is the sets of proteins that fell under that Go pathway, and they are all significantly depleted if you add, if you do a competitive inhibition with a simple ozonide or if you pre-incubate with E64D, suggestive of the fact that these are targets of our peroxide antimalarials. So we wanted to look at this in a bit further. So we decided to establish another way of analyzing whether or not these peroxide antimalarials are affecting the parasite redox homeostasis. So we generated a targeted thiol mass spectrometry-based metabolomics approach in the lab. So where we derivatize all the thiols and we focus on the glutathione pathway in our hand. That's because glutathione is the major antioxidant in our system for the parasite. And what we were looking for was the precursors to glutathione, which I'm showing here, the reduced form being glutathione, and then the oxidized form being GSSG. And we had some controls here as well. So we use diamide, which is completely oxidizing for the cells, and we use DTT, which will be completely reducing. We used our antimalarials, the 3 peroxides, so dihydroartemisinin, the first generation of ozonide, and the second generation. We also use pyrimethamine, which is another antimalarial, and it should not affect redox homeostasis of the parasite. So what you can see that, so majority of our cysteine in our media is in the form of cysteine. So when you add DTT, you increase that cysteine level, but it doesn't really do anything for our other compounds. And then gamma-glutamyl cysteine, which is a precursor to glutathione, was only increased in the presence of our oxidizing and reducing reagent. While glutathione was significantly depleted in our oxidizing reagent, and you could see depletion for dihydroartemisinin, and it looked like it was trending at least for the ozonides as well. So as I mentioned earlier, these ozonides have a significantly longer half-life, which also means that they are slower acting compared to the dihydroartemisinin within the parasite. And then the oxidized form, which I was shown here, GSSG, is increased following addition of the diamide, which is as anticipated. And then you can see that it's decreased following addition of DTT, but it's also decreased with the peroxide antimalarials. Suggesting is that these peroxide antimalarials probably just dampen the redox homeostasis of the parasite overall. Now, so hopefully I was able to show you that the peroxide antimalarials, so the dihydroartemisinin and the first generation and second generation of ozonide targets parasite antioxidant defense. And I was able to show you this using chemoproteomics approach. So we did pull down experiments and also using targeted thiol measurement, which we've established in the lab. We're currently also working towards doing quantification around the thiols as well. And we've got some really interesting data across different lines as well and what that would look like. So in summary, just like to thank everyone in the Creek Lab and Jonathan Benesom's lab for providing the compound, the Archine probes to do these experiments. Thank you.
Thank you. Really cool and so relevant. I mean, we really need good drugs against malaria.
Thank you.
So, if this new compound goes onto market, what's likely going to be the mode of resistance against it?
So that's already been shown in a number of other publications. So there is currently resistance seen against artemisinin in the field, but the resistance is a tolerance more than a resistant mechanism per se. So the parasites, because the drugs are highly active for a short period of time, in that short period, most of the parasite die, but a small percentage survive, and that small percentage surviving is enough for it to come back in the next cycle. And so that's how resistant is seen. And resistant against this drug, those parasites have an enhanced oxidative antioxidant mechanism as well. So they have more glutathione and they have reduced GSSG. So they're highly reduced. And I wonder if that's also a mechanism of them being able to respond to the drug. They also have a number of other pathways, but that's one. And we have tested those ozonides against the artemisinin resistant parasite, and they are cross-resistant. So it's not an ideal scenario, but the fact that they have a longer half-life, we're hoping that eventually it's enough to get rid of those parasites that have just sort of been hiding and are just surviving.
Yeah, that makes sense. Cool. Cool. Do you have a reference for the thioderivatization method?
Yes. So we published that thiol derivatization method in our recent paper. So if you just look up my surname, and you should be able to find it. And if you do peroxide antimalarials, but we did work quite a lot around that, because we were looking into whether or not we should be able to derivatize live parasites, or we need to actually add our derivatizing reagent directly to our extraction solvent. So when we added it directly to the extraction solvent, that was sort of the best way to go forward, rather than derivatize live parasite, because it also caused a lot of other changes in the parasite when we added the derivatizing reagent antigen to those parasites. So there was a lot of stuff that was happening, but we effectively be added to extraction solvent, and then you have some kind of buffering system in there with your extraction solvent. It's enough for it to be activated, and it will derivatize all the free thiols in that sample, and with minimal effect on the parasites per se, or any cells.
Okay, cool. In the interest of time, let's move on. But I'm sure, Giza, you don't mind if Natoya Lloyd, who asked this question, approaches you offline to discuss.
Yeah, no problem. Yeah, thank you.
That's why we love these conferences, right? We do community building, we connect people, and we want to learn from each other.
Yeah, that's great. Thanks.
Okay, cool. So our next lightning talk is by Purva Kulkarni, and Purva is going to talk about precision diagnostics in inborn errors of metabolism. Purva is from the Translation Metabolic Laboratory in the Netherlands at Radboud University. And Purva, over to you.
Thank you. Just a minute.
Oh, I could also say that Purva is the recipient of the latest Indigo Award for female mass spectrometry, sponsored by Indigo. Congratulations. It's been a while.
Thanks for that mention. Just a second. Yeah. Okay, maybe. So I sent you a master presentation just because I got a new laptop yesterday, and it's now asking me to do all the security and privacy things, and doesn't allow me to share my screen somehow.
Yeah, but you have to go to see, do you have it and you want to share it?
Or if
you want to email it to be, you have to go to settings and click all those apps.
Yeah, but it asked me to reopen, quit and reopen Zoom. I'm sorry for taking
no, let's
time.
not do that.
But if the next presenter maybe can go before me, till then I can join the meeting back. That's also fine, if that's fine for you.
Tusi, what do you prefer?
I can share quickly. Yeah. Give me 1 second.
Okay.
Yeah, I'm sorry.
Yeah, Purva, that happened to me the other day as well. I also just changed all my computers. Oh yeah. Man, yeah, all the security settings need to be readjusted. Huh?
Yeah, somehow with doesn't work without that.
Cool. So
yeah,
this is
perfect.
Great.
Could you please make it? Yeah.
Awesome.
Good.
Alrighty.
I will just tell you next, because there are a couple of animations there, but thank you for bearing with me. So my name is Purva Kulkarni. I work at the Radboud University Medical Center in the Netherlands, and today I will talk about my work. So it's a diagnostic lab, and we are doing a lot of omics-related work. And one of the parts that I'm currently or have been working on for quite some time is untargeted metabolomics for the diagnosis of inborn errors of metabolism. And what we do for individual patients. Next, please. Yeah. All right. So we all know that metabolism is very, very important, and it refers basically it's a collection of thousands of complex reactions that take place within a cell. So for proper functioning of these chemical processes, our body needs enzymes, and enzymes mainly help in the conversion of a substrate into a product. So in case when an inherited gene causes an enzyme deficiency, it disrupts the particular chemical reaction that leads to accumulation of substrate to the toxic levels and lack of product. So next, please. So there are a couple of animations. Maybe you can click through all of them. So what happens is once there is an enzyme deficiency, then the substrate goes high to toxic levels, and the product basically is not present, and that leads to a metabolic disbalance in the body. Next, please. So such a chemical reaction can affect a single or multiple metabolic pathways that can alter normal processes, which can lead to many abnormalities. Next and next. Thank you. So what happens then? So such abnormalities are known as inborn errors of metabolism. So these are basically a very large group of genetic disorders. They are rare, but also collectively. So since there are different subtypes, they are not that rare. So it has been found that here in Europe, at least 1 out of 12 people are affected with a certain inborn error of metabolism, out of which about 50% are children. So if these disorders are diagnosed accurately and within time, then they are treatable. So for early diagnosis, as a part of the newborn screening program in many countries here in Europe, but also in Asia, a heel prick blood test is performed, which successfully captures a proportion of the patients, enabling the IEMs, leading to early recognition and prompt therapy. So with early intervention, so the interventions basically can be a restricted diet or supplemented diet or something like that, which the doctors decide. And this diet or the supplements need to be customized per patient. So the newborn programs also use targeted metabolic assays that focus on defined panels of biochemical compounds for selected disorders. So such targeted approaches, however, can limit the number of disorders that can be screened at the same time and face the problem of false negatives as well as false positive diagnosis. Next, please. So with the advent of omics technologies, we have been using quite a lot of whole exome sequencing for this, but this actually gives our answers from a genetic point of view. But if we want to know the answers from a functional point of view, we use untargeted metabolomics for this work. So here, what I will present you is next generation metabolic screening, which is a method that we came up with, which is now being used in our diagnostic lab for screening patients who are suspected to have an inborn error of metabolism. So what we do is we start with patient blood samples that is measured in a liquid chromatography mass spectrometry system, which generates a lot of data. And of course, this is very difficult to process manually because this is untargeted metabolomics data. So we have a computational pipeline, which I will come across in a bit. Once the data is processed, what we get is a complete metabolic profile to look for perturbed metabolites. So currently for diagnostics, we do not use the whole metabolic profile, but we filter we do post-hoc filtering of known metabolites using a metabolic panel. So this metabolic panel basically contains a lot of metabolites that are associated with known inborn errors of metabolism. But we also later use the untargeted metabolomics data, which I will come on in a bit. So once we have once we look at these metabolites, our team of data analysts and laboratory specialists look at these perturbed metabolites, which are basically really very high signals as compared to our quality controls. And this information is conveyed to the clinicians. And based on this, a course of action is planned on an individual patient basis. Next. So here I show you the computational workflow, which was published last year. This computational workflow mainly contains 3 parts: quality control, data processing, and data interpretation. I would also like to mention here that this is already ISO approved and fit for use in diagnostics and goes through a round of audit every year. So the analytical procedure, so the QC procedure basically is automated by using a QC reporting tool, which generates a report that our lab technicians see, which reports the external and internal quality standards in each patient sample with several analytical parameters: retention time, response, and repeatability, and so on. And the computational workflow is basically then is data processing, where we use a lot of different steps. So as you can see on the screen, we use XCMS for peak processing. Once that is done, we perform the annotation of the compounds of the metabolic features using the HMDB database. And after that, we do a lot of statistics, which is then catered to N = 1 patient. And this is the backend, but we also have supplemented this with a seamless graphical user interface, which helps which basically performs all the processing, but also helps the transfer of raw and processed data into our cluster, which makes the pipeline also reproducible even after a couple of years, which also helps because we also look retrospectively into our data. Next, please. Coming on to the interpretation tool, this is like the most important tool that our data analysts and the laboratory specialists use that we have developed and included since the last year in our diagnostic protocol. Basically, what it gives is in a single batch, all the patients that have been measured, you can see all the metabolic features that have been detected and filter these features based on known metabolites, based on unknown metabolites that you would like to explore. And plus it gives you a lot of visual graphics that help the laboratory specialists look at this information right in one screen without using a lot of Excel files as we used to do previously. But apart from that, all the diagnosis is also written down, and we can also chat with the clinicians as well as laboratory specialists using the chat mechanism that we have provided. Plus, we can look at the raw data also in the form of, say, extracted ion chromatograms and so. So this has been a very useful tool, and everything that happens, so even like any clicks or anything that is done by the laboratory technician is recorded in the backend. So all the activity is recorded, and we can always trace back the history. Next, please. So what we are trying now to do on the top is, since we have a lot of untargeted metabolomics data, and we are all the time very much interested in trying to find new biomarkers for known IEMs, but also to find new biomarkers for not yet known inborn errors of metabolism. So in that case, we have now started building a divergent feature database. That's what we call it for now. What a divergent feature database is, we look for different features that are perturbed in also retrospectively, so in our other batches. And so if, for example, in our current session in a patient sample, we are interested in a particular feature and want to see if this feature has been repeatedly found in some other previously measured samples over the last years, we can do that now with the inclusion of the divergent feature database. So this is something that we are developing at our end, but also our aim is to connect that with other university medical centers within the Netherlands to share information as well. So this is for the metabolomics part, but I would also like to say that we are working a lot on other omics like glycoproteomics, and one of the pipelines would soon be coming. It would be soon introduced in diagnostics for diagnosis of a specific set of diseases known as congenital metabolic disorders. But this is my presentation for now. Next, please. And with this, I would like to thank a whole team that has been involved in developing this, who gave a lot of feedback, and because of whom this has been in diagnostics and we are building on the top of it every single day. With that, I would like to thank you for the attention and for the organizers for inviting me, and also for organizing this fantastic conference. Thank you so much.
Thank you, Pova. This is really, it's the future, right? There are multiple questions for you in the chat, and in the interest of time, it would be great if you could answer those. And let's just take Jurek's question, which I think it resonates very, very much with me. How do you communicate untreatable diseases to the parents? And I think the second question, Jurek, is how do you standardize untreated metabolomics? You mean untargeted metabolomics? Yeah, untargeted. How do you standardize untargeted metabolomics to pass through the certification? Because I think you went for 15189, right? ISO 15189.
Yes. I would like to mention that we haven't certified untargeted metabolomics, but we have certified semi-targeted, because what we are using in diagnostics currently is a panel which has a lot of metabolites, and when we look into the untargeted metabolomics data, we slowly add the confident features, the metabolic features that we think are directly correlate. Then we add it to the metabolic panel. So this, in routine diagnostics, we are using a metabolic panel, which is still better than targeted assays. And the untargeted data basically is for a next level comparison. So once we have, say, an interesting feature that we do not know, then it goes through several rounds, say infrared, MSMS, to confirm its identity. And only then we add it to the metabolic panel.
Yeah, makes sense.
I'm with you.
Cool, thank you. Lots of more questions. Can you, in the interest of time,
thank you.
In the interest of time, in the interest of time, we shall move to the next lightning speaker. And actually, Jurek, I think this is the point where I hand over to you, isn't it?
Yes, this is definitely an option. I'm unmuted, and I'm happy to join you from Munich here in Germany. So it's very early morning. I had my breakfast already in the middle of the night. I'm welcoming Romanas Halekis. I hope I pronounce your name. I think you are in Japan right now. At least this is your assignment. If you're online, please show up. I know that you have already logged in. And today we'll be having a presentation about exposome research, fascinating aspect of our interaction between the environment and physiology. Could we have the screen share, please? I know he was among the people who joined already. I have chat with him.
You write in the chat, I do not see option to share. So, Romanas, do you see the big green button at the bottom of the screen? No. Okay. Okay, what we could do is you email your slides to Lucy.
Can we make him a panelist?
No, I think we can't do that.
No,
right?
we can't.
We also cannot hear you.
Anne, can we move to the next speaker while I'm fixing that?
Okay.
Yeah.
Okay.
Sorry.
So we sort it out, and the next speaker scheduled will be Amelia Palermo. Amelia, you are online. I see you. I have seen you previously as well. Could you please set up for your presentation? Okay, this will be on. Right, yes. So please start sharing your thoughts about your project. Welcome.
Thank you very much for this kind introduction and for giving me the opportunity of presenting today. So I'd like to start my presentation with this image. Some of you may recognize here the Vitruvian Man by Leonardo da Vinci. What really impresses me in this figure is the fact that probably this is one of the first attempts in history of looking at our system in an holistic way. And this is what we do regularly in our research by systems biology and metabolomics. Particularly, I'm interested in metabolites. So the reason why is because they are building blocks and energy source for the cells, but also, and perhaps more importantly, they modulate proteins activity and gene expression through RNA, ultimately modulating the phenotype. And interestingly, metabolites are an accessible point of modulation of the system through the diet or the microbiome. Metabolites affect their activity at the cellular level, but what really enables the function is also the spatial expression organization of the active metabolic pathways. So this ultimately enables the functions both in health and disease. But to complicate this scenario, as many of you know in this audience, we haven't charted completely the metabolome. So we are still adding novel molecules and characterizing them. So this is really not a two-dimensional problem, but is more of a multidimensional problem in which we also need to understand protein regulation and transcriptional and genome association. So in this presentation, I would like to give you, to walk you through two different studies. One is about the first aspect that I mentioned, the heterogeneity of metabolism. The second one is about metabolite activity. Starting from the first one, many of you know that we can perform metabolite imaging by MALDI mass spectrometry. This leverages different ionization matrix. They all came with a different bias on the metabolites or the lipids that, or proteins that we can detect. Moreover, the matrix itself generally ionizes very well in the low molecular range, which is the one that we are interested in measuring for untargeted metabolomics. So this is an issue when we aim at comprehensively measuring metabolites over space. And for this reason, we developed a different matrix for performing comprehensive metabolomics over space, which is called the nanostructure initiator mass spectrometry. This leverages perfluorinated gold nanoparticles that, when hit by the laser in the MALDI source, release the fluorinated chains and enable the desorption and ionization of a heterogeneous range of metabolites. In addition, we can also, this also allows to decrease the energy of the laser and therefore reducing the in-source fragmentation with very low interference from the matrix. So we applied this approach for the untargeted detection of metabolites over space using column slices, and finding that for each laser spot we could measure approximately 1,000 metabolic features, of which we could accurately annotate about 80 across very different and heterogeneous metabolite classes, as you can see here. For instance, we could measure at the same time glucose, but also hydroxycortisol and lipids over space. Next, we applied this technology in a comparative study where we investigated the effect of a different diet in mice. So very simply, we put 3 groups of mice in under 3 different diets, a fiber-depleted, a polysaccharide and fiber-depleted diet, or a standard diet, and then we imaged their colon. We report here normalized intensities of metabolites as a sum for each class because of for the sake of space. So intuitively and also expected, what we saw is that reducing fiber and polysaccharides reduces the lumen content of carbohydrates, but also the derived small chain fatty acids. And interestingly, we saw that there was a significant change in the architecture of sulfur metabolism, as you can see here in the last row. So ultimately, as a perspective, we aim at pushing the frontiers of this technology towards single cell and also the integration with other single cell approaches at the systems level. Moreover, if you're interested in the protocol and you would like to try this, we recently made available a description of it. So next, I would like to walk you through the application of metabolomics activity screening for identifying metabolites that modulate the PD-L1 protein. Here, I'm reporting the workflow for activity metabolomics. It all starts from generating the biological samples in the 2 phenotypes of interest that we are interested in modulating. Next, we perform lipidome and metabolome profiling in a very comprehensive fashion using multimodal metabolomics. And then we, through statistics, we isolate the significant metabolites and then we identify them. So a classic metabolomics workflow will end here. In metabolomics activity screening, we go back to the biological model and we test these metabolites for activity. And we did it towards the PD-L1 protein, which is a key modulator of adaptive immunity, particularly highly expressed in cancer, resistant to immunotherapy, using as a cell model acute monocytic leukemia. Very briefly, when a cancer cell interacts with a T cell, there is the recognition of the antigen from the cancer cell from the receptor of the T cell. This activates T cell immunity, release of interferon gamma. However, when PD-L1 is expressed on the cancer cell, this binds PD-1 on the T cell, and this literally turns off immunity. Moreover, the early release of interferon gamma in the microenvironment exacerbates, upregulates the expression of PD-L1 on cancer cells. So it's a very vicious cycle. So in this context, we were interested in understanding which are the metabolites that could modulate the expression of PD-L1. We first profiled the metabolites changing over time in THP1 cells stimulated with interferon gamma. This is because, as I mentioned before, interferon gamma upregulates PD-L1. And the first observation was that we observed the majority of the changes at 48 hours from stimulation, mostly interesting amino acid metabolism, a general increase and a decrease of amino acids that are important in immunity, for instance, tryptophan, and coupled with the increase of neopterin and kynurenine, which are well known and validated markers of interferon gamma activity. Moreover, we also observed the increased lipid synthesis accompanied by decrease of the precursors in this pathway. But instead of really looking into more into this observational data, we went back to the THP1 cells and we tested these metabolites for activity, finding that hydroxyproline and carnitine have a significant effect towards the upregulation of the protein. And particularly in the next step, we focused on 4-hydroxyproline, which, as you can see here, has a dose-dependent effect on the upregulation of PD-L1, which is interferon gamma dependent. This is not only limited to THP1 cells, but is recapitulated also in other in vitro models of PD-L1 expression in cancer, as you can see here. Next, we stepped into systems biology or system pharmacology to gather a better understanding of the mechanism behind the hydroxyproline activity. So we performed RNA sequencing in the presence and absence of the active metabolite with interferon gamma, finding that TNF-alpha and TNF-K pathway, but also the inflammatory response pathway at transcriptional level were upregulated by the addition of this metabolite. And this, in the next steps, pointed us to derive an effect on autophagy. As you can see here on the left, we finally characterized that the effect determined the effect of hydroxyproline towards the upregulation of PD-L1 is mediated by an inhibition of autophagy. And with this, I would like to conclude this part. The take-home message from the second part of my talk is that we have a powerful tool, activity metabolomics, that enables us to chart metabolites that exert an activity towards proteins that are important in disease, such as the PD-L1. And we can gather further insights by combining this technology in the context of systems multi-omics interpretation. And with this, I would like to conclude. I would like to thank you all for your attention. I would be very happy to answer your questions. Thank you very much.
Thank you very much, Amelia. This was a very nice presentation. Maybe I have a one short question to you. This is concerning your research done on colon slices. Are you able to differentiate the conjugated and non-conjugated bile acids along the slices, so along the column?
Mm-hmm. When you mentioned conjugated, you mentioned them conjugated with like second phase conjugation with a
yes, this is what I'm referring to.
Okay. Yes, we could do that to look exactly of what we detected and what we didn't. We should go back to the data and explore them better. But I remember we could see some of.
This is great opportunity. Well, for the sake of time, I would like to stop the live discussion right now. There are some questions in the chat, so please have a look at them and all answer them online. And we are coming back right now. Thank you very much, Amelia. It was a very nice presentation. And now we are coming back to Romanas Haretskis. He was fighting in the background to join us, and he is online. I'm happy to see him speaking. Romanas, please start.
Yeah, thank you for the thank you organizers for having me here, and sorry for the technical glitches. And today I would like to present you briefly my experiences from the developing a high precision and targeted LCMS HELIC method. And I'd like also to show how I came to the conclusion that less is more. So first, each of us have different surroundings, different diets, microbiome, maybe diseases, and all these exposures result in the metabolite profiles in biofluids. And if we can measure those, so then we can quantitatively, then we can probably also conclude to see what were initial exposures and maybe what to avoid or what maybe to enhance. But if you want to measure the exposures precisely, so we need to do it in the biofluids, and there are multiple challenges. So first, there is a range of inter-individual variation in concentrations. So for the endogenous metabolites, it's maybe like 5 to 10-fold in blood, and it's larger in urine. However, when it comes to the drugs or dietary or environmental compounds, these are much more highly variable and often low abundant. This is quite often due to the fact because the exogenous compounds get metabolized and sulfated or glucuronidated, so they can be more easily excreted, so like more polar compounds. And for polar compounds, HELIC chromatography is good, but once we try to measure polar compounds, there are other charged species. And these are inorganic salts, which are often at much higher concentration in the biological samples than the polar metabolites, for example, as you can see here in urine or in blood. And thirdly, when working with the non-homeostatic biofluids, such as urine or saliva, total concentration depends on the physiological stage of the individual. And for example, in urine, as you can see here, samples from a large study, so the urine concentration between the samples can differ 10 to 15-fold. And kind of, so if you want to measure precisely, then we have to kind of normalize or dilute the biofluids to common concentration for LCMS measurements. But how does it combine? Like if you want to really see something low abundant, it's kind of counterintuitive. One would kind of let's try to maximize. And this is what I also often see like in the papers. Let's inject this as much as we can to see the low compounds. And here I would like to present some data which would hopefully will prevent you from doing that. So I took a small urinary sample set, 33 variable urine samples, and in the less is more approach, I have normalized the urine samples, diluted them to the same concentration. Then I extract them with acetonitrile, injected rather small volume. In the max out approach, I just took the urine samples, extracted and thereby diluted them, and then really injected as much as I could to the column, as long as like the chromatography does not break. So let's take a look at the data sets which resulted from this. Well, if you inject more, it's a no-brainer. You get more peaks, you get more signal. Great. So maybe we can see the more also the low abundant environmental compounds. And so is it really better? So let's get down to it. So let's take a look first on the basic data parameters of the data set. So if we look at the base peak chromatograms in the pooled QC samples, in the diluted as well as in the max out approach, in the QC samples, the base peaks overlay quite well, and we see, okay, the max out approach, the signal is more closer to the saturation, but yeah, we wanted more signal, right? We want to see more. And all sample level, then similar picture, we can see the variability between the samples, and we see more signal in the max out approach. When we do a quick all feature extraction and a PCA plot just to see is like the data good, and yeah, all that is expected. Pool QC samples cluster together, and the study samples in both data sets are scattered around, more or less what one would biologically expect. However, there is evil in the detail. And let me introduce at this point, so that my technical internal standards, which I spiked at the same level into each sample, so it's 5 guys. And if I dilute the samples to the same concentration, total solid concentration before the measurement, so I get nice peak shapes, reproducible signal. Well, those compounds are at the same level by the end of the day. However, when one maxes out and really likes to go for maximum signal, this is what happens. We see a massive ion suppression. Some of the internal standards are basically gone, and there is a great deal of variation. If we put this into numbers, so in the diluted urine, we see the CVs of the technical internal standards, and the QCs as well. The samples are very acceptable. However, when we go to the raw urine in the QCs, the CVs still look quite okay if we ignore the two internal standards, which get oversuppressed. But when we look in the samples, it's a disaster if the compound is, which should be present, should be like detected also at the same or at least similar levels, show this kind of the CV. So so when you go for really more, you really lose the quantitative aspect of the individual features in your data. You still see the biological variability, but then kind of you cannot really trace it back. What is the exposure? What is the molecule behind it? Because everything becomes so complex. So, and that I would like to summarize. Well, if you go for as much as possible, yes, one can expect more signal and have a simple sample preparation. However, in addition to all the tear and wear on the LCMS system, we are much less quantitative. And then if we find something, then it's or some feature which shows a distinction, it's very difficult then to follow it up. And yeah, many of the data that are not reproducible. However, if we go for less is more approach, then we get more stable signal. We have a more robust system, so it's the samples do less wear and tear on the LCMS, and we also are more quantitative. So for take-home message, I would like to tell you this. So if you're analyzing samples with variable backgrounds, such as human samples or biological samples, go for less is more approach. Because if you try to really max out, probably you will get more signal, but this will result in spurious qualitative detection of load abundant chemicals, if at all. And I would like to show a couple of slides what is possible when you go for less is more approach. So this is just a glimpse of my 5 technical internal standards on over 1,000 injections acquired in 4 weeks. So this is the raw peak area data without any normalizations, and I do see very little drift. So the kind of and the samples are easy on the instrument, and even for the helic, which is kind of known to be notorious for retention time drift, when you don't overload the column, are really gentle to your system, then it does work. You really get nice alignments, and this really reduces the problems which need to be solved then later down the data processing pipeline. And when talking about the exposome type compounds, so the low environmental compounds, so I'd like to show you just one slide from the organopesticide metabolites, so they'll kill phosphates. And on targeted metabolomics, it's no magic. So when the compounds are present at sufficiently high concentrations, I do see a signal, and it does it does correspond to the data obtained by using targeted methods. However, if the compounds are too low, then non-targeted metabolomics is not the way to go. And I would like to thank you for your attention. If you want to know more about how to get more precision out of your helic method, this is the publication and people involved in this work. And if you have later questions, my email, and I'm also happy to take questions now. Thank you.
Thank you very much. This was a very nice presentation. I appreciate your quiet and precision presentation, despite the challenges you have encountered. I have 2 quick questions. The first one, what is your experience with normalization with 2 different methods, as osmomolarity and creatinine levels? which would you prefer, if any?
I personally would prefer the one which is easier and quicker, especially when going for the large scale. And when you're going for the normalization, like when you're trying to measure the total solutes, so often creatinine correlates with specific gravity and also with the osmomolarity, and then it probably comes down to what is easier to measure. And in my experience, the specific gravity is kind of easiest to measure and can be probably more prone to automatization. So I have automated the specific gravity measurements.
This is very clear answer. Thank you for that. A second quick question is, you shared with us this possible drift of internal standard signal, and I know that it is clear this is a scale which shows that there are some differences. Usually they are very, very small, but I noticed there is a pattern, at least in most of the internal standards you have used. Can you attribute this to any technical issues, or in the middle you have some maybe the second from the bottom is not, but in all others you have some change in the middle? Can you attribute it to column change, pump, maintenance, whatever? What would be the explanation?
Well, this is thank you. Very specific question. System tune. Actually, I do not change columns in the middle of the race. You don't change horses in the middle of the race. So if you have a good method, you can go really for several thousand injections on the same column. I pretest this. So what happened in the middle? System tune.
Okay, thank you very much. This was enough what I wanted to ask. For the sake of time, I'm asking you to look into the chat. There are some questions. Please answer them, and please pass my best messages and greetings to Craig.
Okay. Thank you very much.
Thank you very much. Bye. Now we are moving right now to fascinating field of fault diversity and fault analytics. And our next speaker will be Hina Sharma from National Dairy Research Institute in Haryana in India. I'm looking forward to your presentation. Hina, please start.
Yeah, I'm sharing my slides. So are the slides visible?
Yes, please go on.
Okay, great. Thank you. So good morning, good afternoon, good evening to all of you. So my name is Hina Sharma. I'm working at ICAR, National Dairy Research Institute, Karnal, Haryana, India. So I've been working on the metabolomics since past 2 to 3 years, not much expert like others, but this was
Hina, we are losing your voice. Maybe you can unshare your screen. You could maybe switch off your camera because we are losing your voice. Unfortunately, we have a technical issue.
Are the slides visible?
The slides are visible. Please, this is much better. Just switch off your camera and please go on. So we lost you here on this slide, on the goat.
Oh, okay.
But now the
No worries.
Sorry.
But now
this is
live.
Yeah. Is it the same now?
I don't see you.
Oh, okay.
Still no sharing.
Yes.
Yeah, slides are visible now?
Yes.
My voice is clear?
Yes.
Okay, great. So again, good morning, all of you. So I've been working on this alterations in metabolites of goat milk yogurt during long-term refrigerated storage. So this work was done in US, Oklahoma State University. I visited that university as a visiting scholar under some fellowship. So while goat milk, nowadays we have great demand for the goat milk products because consumers want diversity in their food. They want nutritive value in their food. So goat milk being a nutritive food and by virtue of its health benefits, goat milk products market size has also increased, and it is estimated that over $17 billion revenues will be generated by 2025. And not only in India, we have this increase in other countries also, Asian countries, Western countries also. So, but the main challenge which remains with the manufacturers is the quality of the goat milk products. Because of its lack of alpha S1 protein content, that is the casein protein, alpha S1 casein, that is abundant in cattle or buffalo milk. But when it comes to the goat milk lacks it. And due to this, the fermented milk products such as yogurt or curd, whatever we prepare fermented milk product, that is having a fragile texture. So as soon as you take the bite of it, you will not feel the smoothness or richness in the taste. So that is why this challenge is very difficult. This challenge has to be addressed in order to increase the diversity on the plates of the consumers. So with this, we have planned this work to know the changes in the goat milk yogurt during its refrigerated storage at the metabolite level. So we used untargeted metabolomics approach in this, and we use the starter cultures and stored it at refrigeration temperature 4 degrees. And then the sampling was done at 3 equal intervals: 0 day, 14 day, and 28 day. Usually the shelf life of yogurt is around 28 days. So we have taken the whole storage length. And then these were metabolites were extracted using the derivatization steps and the other cellulation steps, and the sample was injected into GCMS, and further the metabolites were annotated, mapped, and the data was analyzed with the different software tools. Now, what we have found, that total 187 metabolites were there. Definitely, because it was a fermented product, we have so many microorganisms grown in the product, and these are doing the metabolic activity is going on in the product. So we got a lot of metabolites in this, around 187. And out of these, we had 129 which were identified, and carboxylic acids and derivatives were in most abundant state. There 44 classes were found. Then followed by fatty acids 18 and organo-oxygen compounds 15. We had others also, like phospholipids, steroid and steroid derivatives, others. But the major were these. And out of these, we had the differentially regulated also. Those were just 39 in number, with the 22 were upregulated. That is, they were higher in concentration as the storage period increased, whereas 17 were downregulated. So we have performed the differential analysis of goat milk yogurt during different storage time. We have separated these into 3. Day 0 was compared with the 14th. 14th was compared with the 28th, that is the last day. And the last day was also compared with the initial day, so that we can get a clear picture what is going on in the product during the storage. So we have performed PCA for this principal component analysis, where we found the 3 PCs, principal components. However, just to know whether the data was statistically significant or not, we have applied OPLSTA, orthopartial least square discriminant analysis also, and this permutation test. So permutation test was able to, we could able to interpret that the separation of the metabolite clusters, which was displayed by the OPLSTA, that was statistically significant with these R2 and Q2 values. Now this R2 value, it shows us whether this model fit is this of the model fit is, whereas the consistency between the original and cross-validated data. So we were fortunate to have this R2 and Q2 values which were very close to 1. So we had the clear-cut distinction between these two groups here in each and every storage interval. Then we have also seen the exclusive metabolites which are found only in the particular period. So here we could found that only 11 exclusive metabolites were present in between the day 0 and 14. This is the Venn diagram on the left side. And then we had in between the day 14 and 28, we had 13 exclusive metabolites. And that was very interesting to note that day 0 and day 14, that were majorly composed of proteins, dipeptides, tripeptides, and amino acids. Whereas if we go further in the later period of the storage, then we will find more of the fatty acids or carboxylic acids. As you can see here, that upregulated metabolites in between day 0 and 14 were D-alanine, valine cysteine, dipeptide again, methionine valine again dipeptide, phenylalanine again amino acid. But day 14 and 28, propionic acid, pymalic acid, glutaric acid, so all those were there. So this was very much clear that bacteria which is present, or a yogurt culture which was present in the milk, that fermented the carbohydrates first, and they utilized the protein source first, and they broke it down to the dipeptides and amino acids. Further, these amino acids were again utilized to synthesize the fatty acids. So that was the trend which we have noticed in this. We have also processed the data for hierarchical cluster analysis, which categorized these metabolites possessing the same characteristics and recognize the variation between the metabolic groups. So these with the brighter color, they had the higher concentration for the corresponding metabolite here, whereas the lighter one, they had the lower concentration. The next step was to see the correlation between these metabolites, whether the upregulation of one metabolite influences the downregulation, or it also enhances the concentration of another metabolite. So this was the major challenge to see and interpret it in a meaningful way. So here, if we see these two pictures, where the green ones, these are the upregulated metabolites, whereas the red ones are downregulated metabolites. And the bars shown between the two, these two are shows the strength of the correlation. If the bar is thicker, the more is the strength of the correlation. If the bar is thinner, the lesser is the strength of the correlation. So the correlation network also showed that as soon as the proteins or amino acids will increase, fatty acid synthesis will decrease. Whereas if the proteins are decreasing or amino acids are decreasing, means the breaking of these dipeptides or tripeptides, it is enhanced, then the fatty acid biosynthesis will increase. This we have seen in day 0 to 14 and day 14. Then we have again mapped and annotated the different differential metabolites into the TEG pathway analysis, so that we can know what pathways these metabolites are taking for the different synthesis or breaking down of the proteins or amino acids or fatty acids. So here we see in the day 0 to 14, we have majorly vitamin K metabolism. Then phenylalanine and tyrosine metabolism was there. That is why we had the main L-phenylalanine was there, D-alanine was there. Then propionate metabolism was there. Propionic acid was enhanced during the later stages of the period. Then we had valine degradation, leucine degradation, and more of the dipeptides or amino acids were formed. Similar, if we see in the day 14 to 20, we had majorly we had fatty acids also saw the pathway impact was majorly influenced by the phenylalanine, tyrosine, and tryptophan biosynthesis during the initial storage. Whereas as soon as the storage period increases, propionate metabolism was decreasing. So the summary of this study was, it was an untargeted approach, so we cannot see the specific things, but yes, storage interval of day 14 to 28 exhibited higher number of differentially regulated metabolites because of the presence of more fatty acids in that, whereas upregulated metabolites during 0 to 14 days revealed higher proteolytic activity in initial storage period by the bacteria, fermented bacteria. Then a few of the differential metabolites were highly correlated, and we could also see that such studies will help in isolation of certain compounds in future. If we are talking about the nutritive value or health benefits of goat milk, then definitely this metabolomic study will help us to isolate certain functional compounds from the functional molecules from the raw material, so that we can use it in the product. Then improvement in the technological interventions for post-acidification changes also will be there. Post-acidification changes are very much troublesome for the dairy industries. Then it will also pave the pathway for nutritional research for enhancement in fermented goat milk products. So I would like to acknowledge these people. This Dr. Ranjit Ramanathan, he was my supervisor there in the OSU, and NEHEB was a fellowship which I got for this work. Thank you. Thank you.
Thank you very much. So despite some technical difficulties, it was a very nice presentation. We got the messages.
Thank you.
I was too surprised to see the short chain fatty acid metabolite showing up in the goat milk, fermented goat milk. I didn't know that. I've learned a lot. You should know that these short chain fatty acids have been used in Europe as antifungal drugs or additions to bread. Yeah, it stopped in the 70s, and since 70s we have diabetes epidemics in Europe. So coincidence, not coincidence? I don't know. This is a scientific question. You got a lot of questions online. I really would like to ask you to do it and encourage you to do this. One question out of the comments I have seen sent to me and others. What are the next plans? Do you think it's wise to expand the metabolite profile to be analyzed, non-targeted metabolomics, whatsoever type? What are your plans?
So, now what I am thinking to include metabolomic study and targeted metabolomics to see what are the changes happening when the milk is converted to fermented product. Because milk will have totally different metabolites. When the bacteria is acting on it, it will have totally different metabolites. So, and under different processing conditions, because milk undergoes different processing conditions, different dairy unit operations like pasteurization, sterilization. So all of these nutritive value gets lost. So we are going to target how to see that at the metabolite level and control those losses in the nutritive value.
Very interesting. Thank you very much. Thank you so much for the presentation.
Thank you. Thank
We're
you.
switching to the next presenter, Shadrat Mutuku. I hope I pronounce your name properly. We're switching to the different topic. We touched in present session some of the aspects, but we are now addressing the specific lipid distribution in a disease model with mass spectrometry imaging. So Shadrach, please share your presentation with us.
Thank you very much, Professor Damsky. Can everyone see my presentation?
Yes, it's all right. Yes, please go on.
Thank you. I'm Shadrach Mutuku. I'm from the University of Wollongong in Australia, and I'm a postdoctoral research fellow looking at the spatial mapping of complex lipid distributions in a mouse model of Parkinson's disease by mass spectrometry imaging. Parkinson's disease is a progressive chronic neurological disorder that primarily affects motor functions. At a molecular level, mutations in the glucocerebidase gene have been postulated to promote alpha-synuclein oligomerization in lysosomes, which may consequently impair dopamine-producing neurons. Mutations in the GBA1 gene are a major risk factor for Parkinson's disease, and GBA1 encodes the lysosomal GCase enzyme, whose reduced function leads to accumulation of glucosylceramide and glucosylsphingosine substrates. This has possible downstream effects on galactoceramides, lactosylceramides, and ultimate perturbations in ganglioside metabolism. In our study, loss of function of GCase enzyme was by knockout mutation of the GB1 gene at the D409 locus to create homozygous as well as heterozygous models. In our mass spec imaging workflow, I collected 12 micron thick sagittal mouse brain sections from control versus mutant animals on ITO slides, and tissue sections were spray coated with 2,5-dihydroxybenzoic acid, DHB, matrix for positive ion mode mild imaging, or 2,5-dihydroxyacetophenone matrix for negative ion mode analysis. Samples were analyzed on a higher mass resolution Bishop Elite mass analyzer at 50 micron step sizes. This is a picture of the dual electrospray multi-interface, where the first laser beam comes from down here and is reflected through a series of mirrors into the source. And this is the conventional MALDI. And the second laser beam comes through this keyhole window of the MALDI interface and intercepts the first laser parallel to the sample surface. So how does MALDI2 work? First, in simple terms, matrix-assisted laser desorption ionization. MALDI is the detection of analytes using a small molecule acidic or basic matrix that absorbs UV laser energy, causing instantaneous ablation and ionization of molecules in the gas phase. So with MALDI2, the primary conventional laser at a wavelength of 349 nanometers hits the surface. And this is intersected by a second 266 nanometer laser around 400 micron above the sample surface. And this second laser is orthogonal to the first laser beam and interacts with expanding plume from the initial desorption event, causing two resonance photon ionization of the matrix. The charge transfer reactions from the post-ionized matrix to analyte in this temporary reaction vessel boosts the yield of poorly ionized molecules. So whereas MALDI detects sphingomyelin and phosphatidylcholine lipids mainly as alkali salt adducts, as you can see in this top panel here, MALDI2 is quite beneficial for the measurement of neutral glycosphingolipids. So they are mostly detected in their protonated forms with up to 4 to 5 orders of magnitude. And here you can see lipids such as phosphatidylethylamines, ceramides, hexosyl ceramides, as well as hexosyl 2 ceramides, which quite benefit from MALDI2. So I have leveraged this analytical sensitivity of MALDI2 to measure lowly abundant hexosyl sphingosine substrate in brain tissues. And here we can see images of hexosyl sphingosine in wild type as a control versus heterozygous and homozygous mutant brains across 3 biological replicates. And our imaging data suggests potential increase in hexosyl sphingosine concomitant with the mutation status of the GB1 gene, or in other words, loss of GCase activity. As you can see here in this tick normalized box plots from white matter areas of these samples. And this data is further underpinned by liquid chromatography tandem mass spectrometry analysis. We have further gone ahead to develop a quantitative mass spectrometry imaging approach in order to accurately measure these spatial differences. So here we are using an internal standard mixture containing several lipid classes that is pre-coated first on the tissue samples and then followed by application of a MALDI matrix. And this allows normalization of lipid signals at each pixel. In this specific example, endogenous hexosyl ceramides signals are visually expressed as a ratio to the C17 glucosyl beta ceramide. And by doing so, we can see a possible increase in this endogenous species when we look at the white matter areas of the brainstem of homozygous compared to wild type animals. And this is observed in the shorter chain hexosyl ceramides as well as the longer chain species. Shifting gears a little bit, we're also interested in gangliosides, which are a complex class of glycosphingolipids with their synthesis beginning from very simple glucosyl ceramides, then lactosyl ceramides, which then undergoes conjugation with 1, 2, or 3 sialic acid residues giving rise to GM, GD, and GT1 species respectively. To measure these, I've developed an imaging strategy for specially detecting these gangliosides in mouse brain. And here I've developed a method that incorporates 2,5-dihydroxy acetophenone matrix that is doped with ammonium sulfate in order to measure the gangliosides. And this boosts the anionic sensitivity by up to 3 orders of magnitude. Therefore, we've been able to successfully map key ganglioside species in our PD model. And we have uncovered fatty acid chain length dependent distribution of GM1 36.1 in the prefrontal cortex compared to GM38.1, which you can see is in the cerebellum, as well as this midbrain brainstem areas respectively. And this pattern of distribution is also replicated across GD1 and GT species. Other regions we're interested in are the hindbrain and midbrain, where we have also seen differences being exhibited in ganglioside concentrations. So in conclusion, using a combination of mass spectrometry imaging approaches, we achieved precise localization and quantification of glycosphingolipid content in glucocerebidase deficient mouse model of Parkinson's disease. Thank you. I'd like to thank the organizers of VPAP for inviting me, as well as my supervisor, Dr. Shane Ellis, as well as our collaborators in Europe, Dr. Kim Macross, Nathan Hatcher, and Dr. Mikkel. Thank you.
Thank you very much. This was a very clear presentation, a very didactic one. I might have the first question. So different models of PD, of Parkinson, have similar appearance. They different in lipids. This was rather tested for different models, for example, for pink model with brain tissue and not with MALDI. So there's a completely different resolution. You're providing much higher resolution here. Now, I'm very impressed by the possibility of detection of hexazylceramides. This is really going more towards signal transduction rather than towards structural lipids. What is your opinion? When could we assess short-living, lower concentration ceramides in this system? What is required from the analytical point of view?
That's a good question, Adamski. So our method is able to detect hexylceramides and hexylsphingosine, which are very lowly abundant metabolites. And one of the key challenges in this type of analysis or area is to detect bioactive lipids, such as ceramide 1 phosphates or sphingosine phosphates. So there are a number of techniques of tagging these intermediate metabolites. There are a few papers out there, yet is not widely available, such kind of technologies.
Yep, but I'm rather referring as well to your model, because in the heterozygous mice, you are able already to see some small changes. So it's again dosage, definitely. Yeah. And this would be probably from the analytical point of view, the best metrics to test your sensitivity for different and as well coverage for different lipids, because then you have the same metrics, but the challenge is the constitution and changes in the relative concentrations of lipids. So the use of different matrices for ion desorption is one step to go. But what about using some different internal standards, potted together with ionization metrics, just to trigger a little bit more signals and less noise in the areas of interest? What do you think about it?
So that's one aspect of the project we're also looking at. I've not shown that data today. So we have specific standards for glucosylsphingosine, which are either odd chain or deuterated at the SN2 position that we have tested before. I've not shown that here, but this is probably the way to go, because some of these standards are well below the limit of detection of most platforms. So optimizing it using MALDI2 or other laser post-ionization procedures such as plasma ionization, we have the capability or just within the reach of being able to detect. So the key thing is first to detect, then to ascertain what robustly is it exactly what you're measuring using an internal standard. So we primarily use a GHB, which works well with most lipids, but the other matrices such as DHA, which are most suitable for ambient ionization or low pressure sources that we can be able to adopt moving forward.
Okay, thank you very much. So for the sake of time, I suggest we'll go to the next presentation, and I would like to thank Shadrach Mutoku for his presentation. We are switching now to the presentation by Ying Yang, who is bringing to us the fascinating research field of redox oxidation and how this is going to be analyzed in this fascinating world of environment and metabolome interactions as we see it. So, Dr. Yang, the floor is yours.
Okay, thank you. And first of all, I would like to thank the organizer for the kind invitation. I really appreciate this opportunity to share with this community my research. And as you can see from this title, my lab is really interested in cysteine. Well, because among all protein coding amino acids, cysteine is very unique owing to its intrinsically high reactivity and nucleophilicity. Although its abundance in the human proteome is only about 3%, it accounts for nearly 1/4 functional sites on human proteins. So the nucleophilic sulfur ion would allow cysteine to undergo a broad range of chemical modifications, especially oxidations. So for example, you could first oxidize these cysteine ethyl to sulfenic acid, SOH, also known as sulfenylation. This oxidative intermediate can further react with other thiolate to form inter- and intramolecular disulfide bond. Alternatively, it can be over-oxidized to give you sulfenic acid, SO2H, or even sulfonic acid, SO3H. So once become oxidized, most of these oxidations could be reduced by a variety of cellular mechanisms, even if this chemically irreversible SO2H modification can be enzymatically reduced by sulfhydroxide in the presence of ATP. So consequently, these redox changes on cysteine residues can reversibly alter protein functions, just like phosphorylation and dephosphorylation. So once cysteine becomes oxidized in a protein, of course, there will be many different types of consequences. Even just a very small sulfenic acid formation, SOH, could cause very profound consequences. So for example, you can dramatically change the structure and activity of protein. It is quite obvious if there's a catalytic cysteine and you block this cysteine through oxidation, you can no longer do this catalytic reaction anymore. And protein-protein interactions, stability and localization of proteins, all of these and many other types of effects once cysteine was oxidized. So it is not very surprised these redox processes can play a very crucial role in many physiological events like thermogenesis, circadian clock, DNA damage repairing, and so forth. However, when this well-controlled redox network get dysregulated, of course, there will be some adaptive responses, and ultimately there would be some detrimental consequences such as transformation or even cell death. And all these consequences may lead to some major diseases like cancer, diabetes, and neurodegenerative diseases, and so many others. So if we want to know or get a better understanding of whether and how does the cysteine-mediated redox changes affect protein functions and redox signaling in physiology and pathology, we should first know which cysteine across the complex proteome is oxidized or redox regulated, right? However, in a chemical aspect, most of these cysteine redox forms are chemically unstable, and biologically, they can be subject to high dynamic changes in biological systems. Well, like Mike Yoffie, who is editor-in-chief of Science Signaling, said in editorial, the difficulty in this field is largely a technical one. Of course, there are many different type of approaches and a lot of interesting determinants of which cysteine be oxidized in a protein. However, an emergent consensus in this field is to find a way to first stabilize this kind of unstable redox forms of interest, followed by doing any detection or analysis. But the question is how? Here are the traditional indirect chemistry-based approaches for detecting cysteine oxidation. And in this kind of indirect strategy, you can detect cysteine oxidation through the loss of reactivity with cysteine alkylating reagent containing some reporter group. Well, if the cysteine on the protein is oxidized, it cannot be labeled by this kind of reagent anymore. So you could detect this kind of loss of signal when you do any gel-based or Western blot analysis. So alternatively, people in this field would also like to develop some chemistry-based approaches for directly labeling and detecting distinct type of cysteine oxidation. So in this case, if you label the oxidized protein or cysteine with this kind of reagent, you will see an increase of signal upon the oxidative stress. So without any doubt, these chemistry-based strategies, either indirect or direct way, really push this field and expand the inventory of oxidized proteins. However, these traditional approaches do have many limitations. So first and foremost, you could study one protein at a time, and you would always need to use some purified protein or high-quality antibody, which are not always available. And secondly, you cannot localize the exact oxidation sites using this kind of traditional gel-based or Western blot analysis. So in order to get such information, which is quite important for the following functional studies, you may have to generate many signal metagenesis models, which are kind of tedious. Last but not least, most of the traditional labeling reactions cannot be performed in living cells. So my lab is really interested in joining forces of chemoselective reactions and mass spectrometry-based proteomic technologies to analyze cysteine-mediated redox regulations. So first, let's take cysteine in its reduced form as an example. So chemically, it is a very strong nucleophile. So there are many different type of electrophile-based chemistries can be harnessed to selectively label nucleophilic thiol group in cysteine residues. Well, today, the chemical called aldocinamide is still the most widely used warhead for cysteine labeling. Well, perhaps the most known aldocinamide derivative in our field is ICAT, which had been widely used in the field of proteomics for the purpose of quantification, and its original publication have been cited thousands of times. And interestingly, people in the redox field have also harnessed this reagent to develop the so-called OxyCat method for measuring cysteine oxidation. Thus, it is not a very difficult choice for us to also select this kind of classic warhead for chemoselective labeling of free thiol, although researchers, including us, are continuing to train out new chemistry, of course. However, for sulfenic acid, SOH labeling, things are not that easy, because the sulfur item on these modification can function as both weak nucleophile and soft electrophile. But people in our field, many explore its weak electrophilicity for bioconjugation. Well, the first chemoselective reagent specifically for this highly labeled oxiform is a carbon nucleophile called dimidone. So building upon this interesting carbon nucleophile, many others, including us, have ever since developed a variety of SOH detecting reagent. And actually, many of these reagents are commercially available, which really push this field and expand the inventory of sulfonylated proteins. However, actually, the kinetic of this dimidone-based SOH labeling is kind of low. So in the interest of discovering more efficient chemistry for SOH labeling, a classic reaction draws our attention. Actually, this is Nobel Prize winning chemistry discovered by George Wittig decades ago. It has been a widespread approach for the preparation of alkenes through the use of triphenylphosphonyl halide. And this region, also called a Wittig reagent, holds a highly polarized pH bond, which highlights the very strong nucleophilicity of this carbon ion. So we thought we might also use this reaction as this reagent as a reaction partner for the sulfonic acid labeling. Well, to test this hypothesis in our cooperation with Kate Kelly's group at Scripps, we use a sulfonic acid model compound to screen the Wittig reagent reactivity. And very interestingly, monosubstituted derivative, including ketone, ester, and amide, indeed form stable product with a model compound in high yields. So they may serve as a warhead of choice to develop the next generation SOH probes. And I will talk about this a bit later. Well, now let's just jump to the SO2H. Well, as compared to SOH, and this overoxidized cysteine ox form is rarely studied, probably due to the lack of tools for detection. So what's the major challenge for selective labeling of SO2H? Actually, it is to differentiate this modification from high abundant protein thiol. So as compared to this more abundant and reactive thiol, the nucleophilicity of the sulfur in on this sulfonic acid SO2H is kind of low. So we sought to harness the difference in stability of labeling products if you had to label both of them. So for example, we reasoned that a chemical called electrophilic nitrogen species can react with SO2H to yield very stable sulfonamide adduct. And in the meantime, such electrophiles, of course, can also react with more nucleophilic sulfite. But the reaction only gives you a sulfonamide bond that can be rapidly reduced during sample preparation. So with this kind of idea in mind, we actually screen several electrophilic nitrogen species. And long story short, we found one type of compound called daisine always show the best reactivity and selectivity as benchmarked by a model system derived from mutant thiol peroxidase. So in particular, the reaction of a daisine compound called DBAD with protein SO2H can give you full conversion to sulfonamide product within minutes. So we chose this compound for further development. Well, to summarize what we have now, so for the highly nucleophilic thiol, we use a classic carbon electrophile called IAM, aldehyde. So for the unique electrophilic sulfur in sulfonic acid, SOH, we choose a panel of carbon nucleophile derived from Wittig reagent. So for the SO2H, we screen a number of electrophilic nitrogen species with a daisine compound called DBAD standing out. Well, the next question turned out to be how to enrich these cysteine redox forms that have been labeled chemically. So what we can do, so we can do mass spec-based proteomic analysis, of course. Well, just a few weeks ago, this year's Nobel Prize in Chemistry have been awarded to 3 pioneers in developing click chemistry and bioorthogonal chemistry. Very well deserved. And this chemistry or reactions can actually allow for the incorporation of a clickable or bioorthogonal group like alkyne or azide into small molecules or protein modifications, enabling the following detection and analysis. So as such, we could just modify our redox form labeling reagent that I just mentioned to provide a panel of clickable alkyne derivative, including so-called IPM probe for thiol labeling, a well combination for SOH labeling, and a dikine probe for ASO2H labeling. Well, the next challenge is to transform these alkyne tagged protein cysteine modification into mass spec signal for site-specific identification. Here is our working plan. So first, we could choose to digest the probe labeled protein into tryptic peptide, and then do clean chemistry reaction to install some a so-called photocleavable biotin tag onto all probed proteins. Of course, you may notice that there might be some excess unreactive free biotin region, right? And they can definitely compete with the biotin-related peptide during the following enrichment. So how can we efficiently remove them? So as you know, on the acid condition, all these kind of peptide, either modified or unmodified form, can be positively charged. Well, the free biotin region remain to be neutral. So we can take advantage of this kind of difference and use strong cation exchange to remove these excess biotin region, and then we could efficiently enrich all these biotin-related peptide with streptonin beads and do photo release, which are very mild condition to elute the peptide. And then we could analyze the resulting peptide for mass spec identification and site localization. So here's just one example showing you how does a photo released peptide look like. And this is from the ASO2H specific probe dalkine. So as you can see from this chemical structure, this dalkine derivative modification mainly consists of three parts: the probe and ASO2H conjugation moiety showing red, the triazole group in purple, formed by this clean chemistry reaction, and the hexanoic acid tail released from the biotin region that is shown in blue. So you could search these expected modification against the protein sequence database and to identify its modified peptide by mass spec. And it is also important to know that sometimes a probe derivative modification itself is very unstable in mass spec, like this one that can generate many diagnostic fragment ions showing green in this tandem mass spectra, which significantly increase our confidence of identification, because only this kind of modified peptide can produce these kind of ions in addition to sequence ions. So now that we can do chemoproteomics, and the next thing we're going to do is to further validate our methods, particularly in terms of protein-wide selectivity of a test probe. So to this end, we collaborated with computer guys and developed the first assessment tool called PChem for the performance of chemoproteomic probes. Well, long story short, PChem can automatically report high confidence probe derivative modification. And it is specifically designed for probe developers. So it would produce some modification-centric and constructive summary, including accurate mass, precise site localization, probe modifications, and so forth. And PChem can also score the overall performance of a tested probe in terms of their profiling efficiency, modification homogeneity, and also residue selectivity. So lastly, PChem is installation-free program that can be easily managed by any users, even those with no or zero experience in bioinformatics or coding. So in the next several slides, I will show you just one example of how PChem can help. So for the ketone, ester, and almate-based WAM probe for ASOH labeling, the chemoproteomic identification rates by searching expected modification is in accordance with their kinetic rates measured in vitro. Apparently, the ketone WAM probe seems to be the best one, of course, our first choice. However, surprisingly, PChem search allowed us to identify unexpected modification derived from individual probes. And notably, the corresponding identification were far more than those by searching the expected ones. And they turned out to be products with a loss of TPP group via a simple hydrolysis process, at least for the amide-based weighing probe. However, for the ketone and ester-based probe, these mechanisms cannot be right. Otherwise, there won't be an additional 14 Dalton. So based on the accurate mass calculation by PChem, this should be 1 carbon plus 2 hydrogen. Well, what is this? Well, this initial positive outcome could be explained when we realize actually these two probes can actually react with endogenous aldehyde, such as formaldehyde, via classic weightage reaction. And the result alpha beta unsaturated ketone products will actually react with protein cysteine in its free thiol form rather than its oxidized form. So in short, the additional carbon actually comes from the formaldehyde. And it is obvious the ketone and ester-based weighing probe cannot be used for living cell labeling of protein sulfonic acid anymore. Regardless, we didn't see such side reaction for the amide probe. And the good news is these probe outperforms the previous reported probes, including DINE2 and BDD, on almost every aspect, particularly in terms of the profiling efficiency. And the new probe wins by a lot. And so it could allow us to label sulfonic acid in living cells in a much more efficient manner. Anyway, a lesson from these would be like the fastest is not always the best. And we always need to keep this in mind when we develop the chemoproteomic probes. So once we have fully benchmarked the method for mapping cysteine redox forms, we still want to quantify them, which will lead us to dynamics in the context of biology. Thus, we could modify our azido UV cleavable photocleavable biotin region to incorporate a light and heavy linker between the azide and the photocleavable benzoic ester moiety to generate this light and heavy biotin region. So, for example, to compare probe labeled samples generated from two different conditions, like treatment or control, or mutant or wild type, we could just react with them with light and heavy biotin region respectively. After click reaction, we can mix the light and heavy sample together and do the following analysis as the same. So in this quantitative analysis, we could calculate the treated versus control ratio for each detected cysteine redox forms. So let's take this example from our analysis of C. elegans. So oxidation of these cysteine by treatment like hydrogen peroxide could reduce its accessibility to IPM probe, and a lower ratio indicate the increase of cysteine oxidation because it's a loss of signal detection, right? So meanwhile, the treatment induced the formation of sulfonic acid, SOH and SO2H will enable more probe-derived labeling, and it therefore gives you a relatively high ratios. Well, now we could actually globally, site-specifically, and quantitatively profile hundreds to thousands of cysteine redox forms in various model species. So what, you may ask? So next, I'm going to show you one real-world application of our redox proteomic analysis. So for example, in our collaboration with Keith Blackwell's group at Harvard, we globally profile thousands of cysteine redox forms in C. elegans upon accurate peroxide treatment, which will represent several-fold improvement compared to previous reports. And interestingly, we identify many new redox-sensitive events throughout the classic P38 MAPK pathway that is known to be activated in response to oxidative stress. And one of such was functionally validated, that is cysteine 213 Silk-1 protein in this pathway. And this oxidation sensitive cysteine is actually localized within the kinase activity loop, which is critical for activity, of course. Indeed, if we mutate this cysteine to threonine, the P38 pathway could not be activated by oxidant anymore, as measured by this phosphorylation of Silk-1 substrate. And moreover, since Silk-1 is also important for the P38 mediated immune response, we also evaluate the survival rate in wild type and cysteine to threonine mutant worms after the infection with a pathogen. And interestingly, this mutation also increased the pathogen sensitivity, further demonstrating the physiological function of these very specific redox event. Well, in addition to this case found in C. elegans, there are actually many other examples showing that our redox proteomic analysis are able to uncover new cysteine-mediated redox regulation events of functionally important. Okay, altogether, I wish in this talk you could remember that we have developed several set-centric chemoproteomic approaches to globally profile 3 distinct types of cysteine redox forms, including ASH, ASOH, and ASO2H. And in addition to generating high-quality redox proteomic data set, our analysis also provides some interesting leads for functional study. And looking forward, I believe these tools will help us to get a better understanding of cysteine redox regulation in more physiological and pathological contexts. Well, let me end up here by thanking all these hardworking guys in my group, as well as a lot of fantastic collaborators from different backgrounds. And of course, these guys will pay my bills. Thank you for your attention. I'd be happy to take any questions. Thank you.
Thank you very much for your nice presentation, very structured. I learned a lot. Thank you very much for introducing us to the field of redoxome, providing the insights into the chemistry behind the tools, and as well tools help to study it. We are very well behind the schedule. I'm sorry to say that. And
okay.
That's why I'm asking only one question.
Sure.
Do you think it's reasonable to think about some group or maybe single standard, an internal standard, which you could prepare and monitor for the process of redox standard, which would be pre-labeled but metabolically stable, but reflecting the chemistry of the redox molecules you are interested in? It is some peptide maybe. Have you thought about this idea?
I mean, very good question, first of all, and use some internal standard to monitoring these kind of redox changes in biological systems, especially in living cells, is a very interesting topic in our field. Actually, instead of this kind of chemistry-based approaches, actually there are some genetic-based approach that can be used to monitor the redox changes in cellular basis, like some NAD or NADPH probe derived from some fluorescence proteins, and there are a lot of genetic-based sensors that can be used for this kind of purpose.
Okay, thank you very much for this explanation. Thank you very much for your presentation.
Thank you.
I would like
thank you,
to thank all speakers and all participants from the discussion for this session. It was a great session. We are rather behind the time, unfortunately, but this was for the sake of science, so why not? And at this time, I think before we go to the next activity, Tuzi, please remind me, this is the time to show up the awards?
Yes, yes.
Okay.
Do you want to share the screen or?
Yeah, I will share the screen if I'm allowed. Okay, let's do it. I'm sharing the screen. This is the screen I'm sharing, and I have to share, and I have to make it full time. Okay. So in this session today, we have a sponsor for the best lighting talk. The sponsor is Journal of Steroid Biochemistry and Molecular Biology. This is a printed journal, an online journal printed by the Elsevier, published by the Elsevier. The journal is covering the area of originally steroids, but right now rather signaling molecules, with a focus on vitamin D, lipids, steroids, their conjugates, metabolomics, and all chemistry behind it, as well functional physiological changes which are taking place in human disease or in animal models. The journal has impact factor of 5 in this year. It's very well cited, present in Scopus and PubMed. And I'm acting as editor-in-chief of this journal. Today's winner is Giza Siddiqui. Congratulations. Is she online? So the award is granted to you because of your excellent presentation, and we'll send you this certificate through the email. And then we'll contact you with award, which will be transferred from you to you from Elsevier directly. All the best for today. I think that this is all about the this certificate, and we have another one.
Yeah, please hand over to Meng. They will.
So Simon Chang from Westland Omics is here to present the runner-based lightning talk. Please, Simon.
Hello, good afternoon, everyone. My name is Simon Chang. I work at Westland Omics. First of all, I would like to say congratulations to all the owners of the best lighting talks, particularly to Dr. Karani for your excellent research work and extraordinary speech. Congratulations, that's excellent work.
You can introduce.
Okay. Yeah. I would like to take 2 minutes to introduce Westlake Omics to everyone here. So the company is founded in July 2020. We focus on boosting precision medicine and pharmaceutical product development by using integrated technology of prometheus and also computer science, such like artificial intelligence and also big data. So far, we have about 100 people here with all kinds of backgrounds, such like biology, medical, computer science. We also built up collaboration with over 100 famous universities, hospitals, research institutions, and pharmaceutical companies to develop new technology and a new product. We will work hard and also work smart to bring in new technology to improve people's lives. That's all about us. Thank you.
Yes, and maybe they didn't hear us or something. Susie, can you hear me? Susie?
Yeah, I can hear.
So did you hear Simon? Did you hear Simon?
Yeah, we did.
Okay, that's good. Thanks.
Yeah.
Okay, I would like to stop sharing.
Yeah, Susie, maybe
stop sharing. Okay, I'm back. Okay, then we're moving to the next step. And Tiamen, are you online? He's not online.
I'm happy to share the screen, and yeah. Let me quickly go through the. So the Purva is the winner for the Westlake Omics. Is Meng, do you want to say anything else or?
Hello, Susie.
Yeah.
Can you hear me?
Yeah, we can hear you. Yeah.
Yeah, just now Simon told us something about the company. Did you hear that?
Yeah, we did.
Yeah, I think that's our presentation, because he congratulated the winner, and he also talked something about our company and our honor to host this, or to sponsor this event.
Yeah, yeah. Thank you, Meng. Yeah. So the congratulations, Pura. Well deserved. And we can move on to the tutorial then. So you want to introduce?
Well, yes, I can start. This is a new session. The session will be concerning the tutorials, a more practical approach to our understanding tools, which we are using our science. And the first presentation will be from Lennart Martins from Ghent University in Belgium. And his specialty is system biology, which I'm looking forward to your presentation.
Okay, thank you very, very much.
Yes, we hear you.
Let me get my screen shared now, and then we can get started. There we go. There we go. So I hope you can all see my presentation now, and thank you very, very much to the organizers for giving me a chance to talk a bit about metaproteomics, which is a field that we have invested a substantial amount of effort in over the past several years, which is a very exciting field, which is a very interesting field, but which is also a very difficult field. And what I'm going to try and do today is explain a little bit how metaproteomics puts certain specific challenges to people who are entering it, how these challenges translate into real-world problems fundamentally, and how we can solve these problems. So what we are doing today to try and solve these problems in very interesting ways. So in a way, you can think about a metaproteome challenge from a size problem point of view. And by analogy, if you were to look at, say, an analysis of the whole world that we live on as the analysis of a single proteome, you can imagine it's quite a challenge already. And we all know that it's quite a challenge. And we've heard many, many wonderful talks about how we're trying to map that single proteome world. If you then think of the single proteome as a dynamic system, so a system that can be healthy, that can be ill, a system that can be regulated, dysregulated, let's say the complexity grows and the complexity becomes the complexity of a solar system, analyzing a complete solar system. However, if you look at the microbiome, that complexity becomes the scale of a galaxy. It really is that kind of scaling that we see happening when you go to entire microbiomes, because then you're suddenly looking not at a single proteome, you're looking at very many different proteomes, all of which are dynamic. Even within a single species, the dynamics can be different from individual cell to individual cell. And then they all interact with each other in very interesting ways. And if these microbiomes form part of another organism, like say human gut microbiota, then the host also plays an enormously important role in regulating that system. So it becomes extremely complex in a way, which is of course where most of the challenges come from. And we'll talk a bit about some of these challenges because we don't have time to talk about all of them and how to solve them. So the way we're going to go through this is we'll first look at the key challenge in proteomics identification in general. It's not specific to metaproteomics, but I will use this to show you that in metaproteomics, this identification problem is especially difficult. And then once we've done that, we'll have a look at a benchmark study that was published recently, where a lot of labs got together to kind of do a state of the art right now at that time of metaproteomics and to see what the effects are of these challenges and what metaproteomics is already capable of at the moment. And then we'll look at some solutions to the fundamental problems. This is very new stuff that we have been doing in my lab very, very recently. And that is trying to solve the identification issue with machine learning. And very finally, I will briefly speak about a metaproteomics initiative that is a global endeavor that started within the European Proteomics Association, which could be relevant to anybody who's interested in metaproteomics. So let's first look at that key challenge in proteomics identification. What you can see here is a theoretical spectrum for a peptide with the apt sequence peptide. It's not a triptych peptide, obviously, but it's an example. And what you can see is you can see all the B ions here. And ideally, if we would live in a perfect world, this is what a mass spectrometer would give us. Because this looks a lot like nucleotide sequencing in the sense that we can just jump from 0 to the first peak and figure out that the mass delta is a proline. And then we can jump to the next peak and find a glutamate. And we can jump to the next peak and we find a proline again. And then we find a threonine, and then we find an isoleucine or a leucine. And then we can jump and jump. And what we are effectively doing is we're ladder sequencing the peptide. Of course, there's not just B ions in proteomics, there's also Y ions. So this gets a little bit more complicated because we don't know which of these peaks represents a B ion or a Y ion. So every time you jump from 0 to a point, you don't actually know which peak you should jump to stay in the sequence. So you could jump to the proline peak here, the B1, and then do you jump to this one, which is the B2, which would be the good choice, or would you then jump to this one, which is the Y2, which is the bad choice? And so that would complicate matters, but only just a little bit. We could still ladder sequence it because the sequence is present in 2 different series here on the spectrum. Now, of course, this is not realistic. A real spectrum looks something like this. What can you see? You can see that quite a few of the peaks that we expect are actually missing. So we don't get full sequence coverage of the peptide. The other thing is that there's these red peaks everywhere. And these red peaks are peaks that come from something else. They are peaks signal that is not derived from the peptide analyte that we are trying to identify. This could be spurious peaks from the instrumentation. It could be spurious peaks from something in the sample, or it could be another peptide that co-fragments and so-called chimeric spectra. Spectra that contain more than one peptide at a time. The biggest problem, however, is the missingness of the peaks, because what happens now is we can still jump from 0 to the B2 ion, and that is giving us some information, but it is only giving us very ambiguous information. So while the total mass of these two of this peak could be represented by 2 amino acids, in this case a proline and a glutamate, it could of course also be a glutamate and then a proline, because there's no difference at the mass level between these two. Essentially, a single peak gives you a composition, not a sequence. However, it could very well be that 2 isoleucines or 2 leucines combined give you the same mass as a glutamate and a proline. And that means that all of these now become possibilities. And of course, since leucine and isoleucine are isobaric, they have the same mass, we can also have all possible substitutions thereof. So what you can see right now is that there's many possibilities to fit this gap. Then if we're lucky, we jump to the B3 ion, we see a proline alone, so that's wonderful. We now know which amino acid is here. But then when we jump from the B2, we have to jump to the B5 because the B4 is missing. And again, we have the problem of ambiguity because multiple combinations of amino acids might fill this gap. Ultimately, when you combine this, instead of reading a sequence, what you get is you get a combinatorial explosion of all possible combinations across these potential sequences. And so rather than say this spectrum represents this peptide sequence, you are now saying this peptide represents one of the following list of possible sequences. And of course, that is not very useful for people. And people want to have a clean identification of a spectrum. They want to know which peptide and henceforth which protein is there. They do not want to see a whole list of potential peptides. So this is the fundamental problem of proteomics, is that we do not have true sequencing capabilities. We have information on the sequence, but the information is incomplete. The way that we resolve this in proteomics is that we kind of cheat. We cheat because we say, rather than expose all possible sequences that would fit this spectrum, we know something about the sample. We know, for instance, that the sample came from a rice plant. And if the sample comes from a rice plant, we're going to assume that the proteins present in the sample are rice proteins. And we're not expecting, say, human proteins. We're not expecting Arabidopsis proteins. We're not expecting E. coli proteins. And therefore, instead of trying all possible amino acid sequences, we will restrict ourselves to only those that come from the rice genome. And that is an extremely powerful filter. It's an extremely powerful filter, and I will illustrate that with some basic numbers. If you take all 10 amino acid peptides, which is just a very small subset of all possible peptides, then you already see that you have 20 times 20 times 20 times 20, so 20 to the 10th number of possible peptide sequences that could fit that spectrum. That is a very large number. I do not actually know how large that number is, but it's ridiculously large. If you then take, say, all the proteins present in the rice proteome, you will probably end up with around about, say, 2 to 3 million peptides. And 2 to 3 million is a lot less than 20 to the 10th. So what we are effectively doing is we're saying, rather than expose ourselves to the full sequence space, we will limit our sequence space to a very, very small, tiny little subset. And that resolves this ambiguity problem. That resolves the fact that we have so many options, because now we can say, of all these potential options, only the one that has EP, P, and TLD occurs in my database. And all the other options are therefore wrong. That's the way we fix this problem. And so in a way, I always joke that in proteomics, we are addicted to databases. We need that database to resolve our ambiguity problem. And the way that we do this, of course, is we take a database, we then mimic what we do in the experiment. So we do an in silico digest with the enzyme, which is typically trypsin, but it can be another one. This will give you a long list of peptides. For a typical eukaryotic organism, you can think about between 1.5 and 3 million peptides. For a bacterium, it will be a little bit less. It will be more on the order of 500 to 800,000 peptides. And then you do an in silico MSMS, which is very similar to what I showed you here. This is an in silico MSMS effectively, because we can calculate all the masses. And then we get theoretical spectra. And then we take a real spectrum, match it to the theoretical spectrum, and somehow mathematically we calculate a score for this match. Then we can rank all the possible hits based on their score, and then we choose the highest score, to which we apply false discovery rate calculations and so forth to make sure that we don't pick too many wrong ones. And so that's fundamentally the trick that we use, but it all starts with the database. While we do this, however, we make some very important assumptions that are not obvious unless you look for them. The most obvious one that is represented on this slide is when you look at the theoretical spectra. The theoretical spectra do not look like real spectra because we assume every peak to be equally abundant here. We effectively make absence-presence spectra. So we say there is no peak, there is a peak. There is no peak, there is a peak. There is no peak, there is a peak. And we do not say this is a tall peak or this is a low peak. That is a very silly assumption in a way, because real spectra don't look like that. Real spectra have different peak heights for their peaks. The other assumptions are a little bit less obvious, one of which is that all peptides here are considered equally probable, which is also not true. So when you digest a proteome, not all peptides will be seen. Some of them are too hydrophobic. Some of them might not even be made at all because the cleavage point where trypsin should cleave is for whatever reason not very accessible. And so we know that we don't get full peptide coverage. Nevertheless, we will calculate all peptides. So there's all these little assumptions in here that we know are violated by reality. Nevertheless, the approach works really well and has been working for very many years for the field of proteomics, because in the end, we still have enough of a filter to make the system work. So why am I making such a big fuss about this? Because it goes very, very wrong when we go into more difficult situations. So what is a more difficult situation? Well, most of the interesting ones. So this is, for instance, the size of a database represented for, say, the human complement of UniProt, KB, SwissProt. But when you do something like immunopeptidomics, which we will hear about in the next talk, proteogenomics, of course, metaproteomics, which we're talking about here, or open modification searches, effectively, we're making that filter that we had, that very small database, we're making that database a lot bigger. In immunopeptidomics, because we have to take into account non-tryptic cleavage. In proteogenomics, because we have to take into account many more potential sequences. In metaproteomics, because we deal with many different organisms, and so therefore we don't have one proteome, but we have very many proteomes. And in open modification searches, because we allow all the amino acids to become very different things all the time. And what happens when we use these very big databases is our system starts to break down. So we now have a very large database instead of a normal one. And the direct consequence of that is we have many more peptides. So instead of 1 million or 3 million peptides, we now have 400 million, or we have 1 billion peptides. So the scales really go up quite dramatically. That means we also have a commensurate increase in the number of theoretical spectra, which all start to look alike. Well, they don't all look alike, but there's going to be so much diversity in these spectra that in the end, we're going to have a bunch of peptides that are not exactly the same. You can see these are not exactly the same, but they look alike. And that means that the scores that we assign between matching their theoretical spectrum to the real one will start looking alike as well. And this is a really big problem. We get non-distinguishable peptide scores. Fundamentally, what we're seeing is the problem of ambiguity just comes back because our filters are not very good anymore. We now have a problem that our search engines say, these all look the same to me. So rather than one peptide, I now have to give you a list of peptides because I cannot tell them apart. And I will not do that because you have told me to give you a single peptide. That's what you want. And the result of all of this is that your identification rate goes down because there are too many of these spectra that do not contain enough peaks to distinguish between these possibilities. And it's the same for all of these problems. How big is this problem? Well, we actually, in a very nice review, we went really deep into this. We redeveloped Extendum so that we could quantify the effect of this. This is not for metaproteomics, but it comes close. So you see the effect on the number of peptides you have to consider for a given precursor based on the settings of the search. And this is the default search. Say this is you search a human database with standard settings. So this is baseline. This is a good situation that we have under control. Notice that this is a log scale, a log 10 scale. The highlight that I put here is when you look at all vertebrates. That is similar to a metaproteomics problem. And what you can see is that the number of precursors you have to consider is vastly larger. It's about 10 times at least larger for all vertebrates. But all vertebrates are actually evolutionary relatively close to each other, which means that there's going to be quite a bit of overlap in the peptide. So the unique sequence space is not expanded that much. If you go to microbes, that sequence space will expand more because there's typically a higher diversity in their sequences. So that will make the problem even worse. What is the effect of this increased search space? The effect is that your identifications here for vertebrates versus the default, your identification rates will drop, especially at the stringency level at 1% FDR. You can see here what the effect is. The effect is quite noticeable. The effect gets less noticeable as you go to 5% FDR and when you go to 10% FDR, which is one of the reasons why in metaproteomics, people tend to search at 5% FDR rather than 1% FDR because the effect is a little bit less. And it is quite noticeable. Now, let's have a look at some metaproteomics data to see how big this problem is there, because I told you that the vertebrates is one thing, but microbiomes tend to be even harder. So let's have a look at the actual impact and see what the results are. So this is a study from 2015 where we did a comparison. We used Pyrococcus here as a model organism. Pyrococcus is an extremophile that lives at the bottom of the ocean near hydrothermal vents. It's a very special bacterium. It requires a very large amount of pressure, about 50 bars and above, and it grows at the temperature of 112 degrees Celsius, which is quite crazy, which means that the proteins it has are quite crazy too. And that also means that the sequences of this organism do not look like any other sequences for, let's say, normal organism, as in not extremophiles. What you can see is when you search Pyrococcus against its own database, you get a certain result. This is a typical, I have a proteome, I search it against the species-specific proteome database. So this is a typical proteomic search. When you search the same data in a database that contains Pyrococcus, so it contains the single proteome, but it also contains the human intestinal metaproteome database. So now it's searched against a very big database with a lot of microbes in there. You can see that the identification rate at 1% FDR essentially gets cut in half. And it gets cut in half on the spectrum identification, so the peptide to spectrum match level, as well as on the peptide sequence level. In fact, the peptide sequence level gets an even slightly bigger hit. As I showed before, for 5% FDR, the effect is a little bit less dramatic, but still it's pretty strong. So here you don't have half, but it's more like 40% to 35% hit on the identification rate. And this was one of the reasons why people in metaproteomics chose 5% FDR, because at least you attenuate this problem to some extent. We can go a little bit deeper. This is the similar analysis now with multiple search engines. So we have Extendum and we have OMSA to compare what happens. The full lines here is what happens when you search a single proteome database. What you can see is that OMSA at 1% FDR, this is 1% FDR, OMSA is not as good as Extendum here. OMSA is an older search engine and Extendum pulls some tricks, so that is not completely unexpected. And when you combine the two, which you can do, you can combine the results of two search engines in a clever way, then you can see you don't really get a dramatic gain over the best. You still see some gain because some identifications by OMSA will have been missed by Xtandem, and of course vice versa. And so the combination of that is a larger number of identifications. Now, what happens when we search, and this is the dashed lines here below, when we search the large database? First and foremost, you see the effect we talked about earlier. We go from about 9,000 identifications to about 4,000 identifications. So the identification rate gets cut in half. That's really bad. The other interesting thing is that now the two search engines have very similar performance. That is because some of the tricks that Xtandem exploits are effectively broken by the very large size of the database. The other thing that you see, and this is actually quite interesting, is that the combination of the two search engines now gives you a better yield. So the delta here is much larger than the delta here. Why is that? Well, when it gets tough to identify things, when ambiguity really matters, the difference in scoring engine between Xtandem and OMSA, which gives them slightly different results, becomes more beneficial. So when the problem gets really hard, it helps to throw multiple algorithms at the problem because each of these has their own little sensitivity and sensibility, and that yields ultimately a better result. We will see that again confirmed later in the benchmarking study. Nevertheless, if you compare the orange line here with the orange line there, you still get about a 50% hit on your identification rate. So while this helps, it's not really making you very happy either. It's not brilliant. So we still have this problem. Where does the problem come from? Well, you can see where the problem comes from when you plot the target hits and the decoy hit score. So the decoys are, of course, what we use for false discovery rate calculation. And in the Pyrococcus database, so the single proteome database, you can see that the Pyrococcus hits are nicely distributed with bad hits and good hits, and that the decoy hits are very low in score, which is what you expect because they're model random hits. However, when you go to the large database, the scores for Pyrococcus move a little bit, but not by much, because the peaks are still at the same place. But now you see that the decoys have shifted to higher scores. That is because ambiguity settles in. There's more sequence diversity, therefore there are more peptide sequences that look like Pyrococcus sequences, and they become difficult to distinguish. So effectively, the good hits are here, the bad hits are here, and the good hits are now starting to move into the bad hit, and the bad hits are moving into the good hit territory, but the good hits are not changing. And so that is fundamentally the problem. Some people have suggested that two-stage searching would be something to do, where you do a first search against a small database, and then you take everything that is not identified and move that to the larger database, which means that you can use the small database to get a big bunch of identifications early, and then try to do some additional identifications in the larger database. So these are the two screens we saw before, with the decoys shifting to the higher scores. So what happens in this two-stage search? You can see that the decoys have not moved so much, and that your targets, they also stay where they are. So fundamentally, this seems like as if it solves the problem to some extent, and it increases the identifications. However, you're messing with the fundamental assumptions behind the statistics of false discovery rate. And you can see the effect when you look at this. When you look at the normal Pyrococcus database, the amount of Pyrococcus sequences we identify obviously is 100%. There's nothing else to identify. So this is our baseline identification, and it's all Pyrococcus sequences. If we search the larger database, then what you can see is that we halve the identifications. We talked about that, right? So, but 99.4% of the sequences are Pyrococcus, which they should be. They should not be anything else because the data is Pyrococcus. And you see that we control the FDR at 1%. In fact, we're a little bit pessimistic. It's the result of the larger database that this thing actually becomes more reliable. So we make an error on the order of 0.6%. So rather than 1% FDR, we have 0.6% FDR. So we get low identifications, but they are reliable. And at the peptide level, you see that, again, you get the same cut, but then the reliability goes down a little bit. It's because you aggregate errors. It's a typical effect, but it's still well controlled. You've got 1.5% FDR and you expect 1%. It's still okay. When you do the two-step search, however, things look better because now you seem to have rescued the situation. You have nearly the same number of hits as the original, but the number of Pyrococcus sequences is only 91.5%, which means you're effectively looking at an 8.5% FDR, which is 8.5 times larger than you think you have. When you go to the peptide sequences, the situation becomes dramatic. You actually identify more sequences than original, which is very suspicious in its own right, but your FDR is now close to 20% at 1% FDR. So you're underestimating your number of false hits by 20%. If you look at 5% FDR, which is where most metaproteomics studies were done, this effect gets worse. Let's just focus on the end result. You seem to get a lot more identifications, but you have an FDR of 40%. So two-stage searching looks good in the numbers. It is not reliable at all. Yeah. So whenever you try these kind of tricks to solve a very difficult problem, you're effectively shooting yourself in the foot. You have to be very, very careful with this kind of stuff. Let's have a look at how bad this problem is. So when we do a big benchmark study, can metaproteomics deliver? This metaproteomics study was called the CAMPI study. It was published in Nature Communications, and it's a very large consortium of people who have analyzed a certain set of samples in very, very many different ways to try and see what happens when you do that. So fundamentally, there's two types of samples. There's a mix that was lab assembled. So you kind of know which bacteria are in there. You don't know how many of each there are because it's an ecosystem, but you know which bacteria you inoculated into the original sample. So you know the mixture composition at least. And then you have human feces, which is a human gut microbiome, where you don't really know the right answer. And then people did lots of different ways of disrupting the cells, fractionation, digestion, cleanup, separation, ionization, and mass spectrometry. And then they also used a lot of different search engines to get to the results with lots of different databases. And the idea was, let's compare between all of these different ways of doing things, and let's see what the effect is of all these differences on the final result. Because ideally, you want this to be robust. You want to be able to say, regardless of the path I take through this analysis pipeline, I get similar results. That's the key thing you want to achieve. Now, when you look at the differences, you can see that the number of spectra, which is always in red, the number of spectra differ quite a bit between the different approaches. Some approaches and some instruments yield very many more spectra than others. There's also differences in the identification efficiencies, which also differ between different approaches. And they're on the same order-ish, but some are a lot worse than others. And so this yields actually quite a bit of a difference. And the difference is even more pronounced in the real life human gut microbiome samples than in the lab assembled samples. So here the differences are even larger. So that did not look very promising, because you look at this and you say, oops, we might be in trouble. However, these are the different methods that have been applied to the analysis. However, when we look at the different bioinformatics pipelines, we also see that matters quite a bit, which is not unsurprising. You see here, Metaproteome Analyzer is one tool, you see Proteome Discoverer, you see MaxQuant, and you see Peptide Shaker. What is relevant here is that Peptide Shaker is using multiple search engines. It's using 4 different search engines. The result of which is that you get the most identifications with Peptide Shaker. This is what I showed you before. If you combine multiple algorithms in a difficult situation, that will give you a boost. And you can see that here, which is the overlap between all of them, the overlap between combinations of them, and this is the unique contributions. And you see that most of the unique contributions come from Peptide Shaker, followed by MaxQuant, and then very few by Proteome Discoverer and MPA. And if you look here this is the fecal sample, and this is the lab assembled mix. In the fecal sample, this effect is much more pronounced even. So the harder the sample, the more difficult it is, the more you gain by using multiple search engines. It's a very simple conclusion. However, the good news is that a very substantial proportion is overlapping quite well. It's a little bit less for the complex sample, but that's mostly due to the fact that Peptide Shaker is so much more effective because it has multiple search engines. If you would take this unique hits out of the picture, that number would be much larger. So one is clearly better than the other ones, which biases this a bit. But fundamentally, they agree on a lot of the identifications, and that is a really good sign. Also, once you go to the protein subgroup level, and when you say, which kind of things do I identify? So here you see unique peptides, and there's quite a bit of difference between the different samples. And these are different samples, and you can see how many identifications there are in terms of unique sequences and peptide level. There's huge differences. But when you look at the proteins that you can extract from that, these differences are smaller. And that is actually really good news because it means that at some level they find the same things. And most of these additional peptides go into better coverage for the underlying proteins. That's really nice, this better coverage, if you want to do, say, quantification, because you have more data points per protein. But it also means that in the end, they are looking at the same proteomes and they're identifying the same proteins. Only some do it with 5 peptides per protein and others do it with 1 peptide per protein. If you look at the top 50% of the identifications, then they overlap very, very substantially. There's practically no unique stuff anymore. So the high abundant proteins, they're found by everybody. It's the less abundant proteins when you start seeing the differences, that is as expected. A little side effect of this was that we came up with a tool to do protein inference in metaproteomic samples, where protein inference and the delineation of proteins and protein subgroups is very difficult. I don't have time to talk a lot about this, but if you do metaproteomics, it's worth having a look at this tool. It's freely available and it's easy to use, and it might actually help you get these more consistent results across your data analysis. So it's just a little shout out there. If you then look at the functions of these proteins, the overlaps become even more impressive. So this is Pearson correlation on the top. This is Spearman rank-based correlation at the bottom. And so fundamentally, between the different samples, you see that the protein-centric functional correlation is extremely high. And that is really good because that is where metaproteomics is used. It's used to find out which are the active functions in the samples at this particular moment in time. And these functions, regardless of the approach, and despite the fact that these approaches identify vastly different amounts of peptides, they actually are extremely similar. So to get a functional representation of a sample, metaproteomics is a really good technology. That was really nice to see. It is a very robust technology. And if you only care about finding these functions, it does not really matter what you do, you will be able to find it. If you want to do very detailed quantification, of course, you want to use technologies that give you a lot of spectra. But if it's functional level, then they're all equivalent. We see the same thing with a gene ontology study with MegaGo, and that is another shout out. So we have made this tool called MegaGo, which allows you to combine multiple omics data sets, and they can be transcriptomics, metabolomics, and proteomics, and do Go analysis across these in a very interesting tree-based way that uses information propagation through the trees. It's a long story to explain it in detail, but the bottom line is this is a really cool way to compare multi-omics data sets at the Go level. And so again, it's really available, it's easy to use. So if you're interested in doing this, either in the metaproteomics or meta-omics context, or in a multi-omics context for a single organism, this is a nice tool to play around with. But I won't say too much more about it here. So what we have learned so far is metaproteomics is difficult because you lack identifications. Different technologies can yield very different numbers of identifications, but ultimately at the functional level, at least these things look very similar. So the technology works if you want function. But of course, we will probably want to do quantification and we'll want to do deep dives into metaproteomes in the future. So we probably need better identification technology to bring the level of identifications in metaproteomics up to the standards of what we are used to in normal single species proteomics. And for that, my group has been working for the past 10 years on machine learning approaches to make that better. And the result of this is MS2E score. So it builds on 2 technologies. The first technology is MS2PIP, which is a fragmentation predictor. This is a real fragmentation spectrum as shown in peptide shake. And this is the predicted fragmentation spectrum from our tool. What you can see if you say focus on the less busy part here, which is easier, you can see that the predictions are actually very accurate. Here you can see overall prediction accuracies for different types of instruments. Bottom line is, yes, we can predict these things very well. Here we make the difference between a TMT and a non-TMT model to show you that if you do special chemical modifications, it makes sense to build a special predictor. This is HCD fragmentation. This is the real TMT spectrum. You can see there's a lot of difference. But if you build a predictor for TMT spectra, you get a beautiful correlation. So that's why you have to build sometimes special models. But we can do this. Why is this so important? Remember this picture, remember the problem that we have here? This assumption here of every fragment ion being equally likely, we can now get rid of that because we can now predict what the actual spectrum will look like. And we can now use the intensity of each of these peaks as a means to score. And the result of that is that we will be able to see the difference because by changing the amino acids, the intensity of these peaks will respond. The bonds here change. the strength of the bonds change and the fragmentation profile will change, not in the mass so much, but it will change in the intensity dimension. And that is exactly what we will be able to pick up with these in silico predictions. At least that's the theory. We'll see in a second whether it works. The other thing that we can predict is retention time. And so we predict retention time with DeepLC. DeepLC has a special trick. It does not only predict the retention time, it can predict the retention time for modified peptides. That's something that we use in open modification searches. It's not that relevant yet for metaproteomics. And I won't talk too much about it, but I'll just show you this one little effect that will show up later in a different way that we did not expect. This is what happens when you predict the retention time of an acetylated peptide if your algorithm does not understand modifications, which is all of the retention time algorithms except DeepLC. What happens is they predict the retention time a little bit too early because the acetylation makes the peptide more hydrophobic and the algorithm is not aware of that. So the predictions are on a nice line, but they're a little bit offset to the real time. If you use DeepLC, it knows about modifications and it can accommodate them. You can see that the accuracy changes. So now it moves back onto the line. That's good. That's what we expect, that it would be more accurate. However, there's a more subtle effect. Look at the spread on the data. This data is spread pretty widely. The DeepLC data is much more narrowly spread. So what you get is you get an accuracy change, but you also get a specificity change. So the thing becomes more reliable, more specific. And that is something we had not expected, but it shows up everywhere. You can see that here in the size of these box plots as well. The blue box plots get shorter, get thinner, which means that you actually get accuracy and precision. You get both. What happens now is that we are going to look at standard search results where the target and decoys are used, and we're going to use a percolator that is now informed by the search engine features like a normal percolator machine learning algorithm is, but we're going to give it MS2PIP features and DeepLC features. So we're going to give it the features that predict the analyte behavior, and we call that approach MS2rescore. That tool is freely available. You can download it here, and you can see the papers here that are underlying this. And the idea is that we make a better separation between the bad hits and the decoys and the good hits. So what is the reality? Oh, sorry, just this is the interface of MS2rescore. So the URL again, it's freely available again. It's very easy to use. You can take your search engine output files and there's many that it can read, and then it just automatically spits out better results for you. So how much better are these results? Well, it's quite impressive. If you take 3 different metaproteomics data sets from very different origins, and you look at what happens with the identifications, this is the baseline you get with Xtandem, the search engine. This is what happens when you do Xtandem plus percolator. So percolator really helps, huh? It's very useful. This is what happens when you use MS2rescore. So the gains are quite substantial. And it's in every single type of data set. This is human gut microbiome. I think this is a biogas plant. And this is a soil sample. Soil is not very, very difficult because we don't have the genomes for most of the soil bacteria. We just don't know what's there. But you can see the gains are always very dramatic. And in fact, we did a little study on the side where we got rid of the Xtandem stuff and we do it only with the predictions. And you can see we get a little bit of a hit. So it's slightly less performant than when we use the search engine, but effectively we're still beating everything. So we don't need a search engine anymore. We can do this only based on the predictions and the machine learning if we want to. This is even more interesting. This is what happens when we look at the Campi mixture and we check what identification rates we get at different percent FDR. So this is 5% FDR, 1% FDR, 0.1% FDR. Same kind of things. This is the classical search engine. In this case, it is MaxQuant. And MaxQuant does not even reach 1% here. It just cannot give you results at 1%. That's why, again, metaproteomics people tend to use 5% results. Percolator makes it better in all 4 of these samples. It makes it quite a bit better. And percolator allows you to go down to 1%, and you can actually get more results with percolator at 1% than you would have gotten at 5% with MaxQuant, so that's good. However, when you use MS2rescore, you can go well above that, and the hit you take when you go from 5% to 1% is much, much less than the hit you would get with Xtandem, and that is consistently the case. What is even more impressive is that with MS2rescore, remember I said with DeepLC, you get accuracy gains, but you also get specificity gains. You see exactly the same thing here because we can go down to 0.1% FDR, which is completely unheard of in metaproteomics. And we still get the same kind of results you would get from percolator at 5% FDR. So you are making no sacrifices. but you're getting 50 times more reliable data. That is incredibly impressive, and we had not expected that. We had expected this, we had not expected this. And it's very consistent across the analysis, always the same picture. I know the next talk is about immunopeptidomics. We have exactly the same results for immunopeptidomics. The plots look exactly the same. So this algorithm can also do that difficult challenge in exactly the same way. And for those of you who do single-cell proteomics plots look exactly the same as this. This is a very, very powerful approach, and you can apply it to your data today. So it's well worth checking out. One of the interesting things is downstream, what is the effect of moving from 1% to 0.1% FDR? Does it make sense to go to that level of specificity? Well, it does. This is Unipept. Unipept is another tool we developed in Ghent that I don't have time to talk about in detail, but check it out. It takes a list of peptides and it plots the taxonomy of that list of peptides at the lowest common ancestor level. It's a really, really cool tool to do phylogeny on your metaproteomics data, and it's ridiculously easy to use. Just come in with a peptide list and everything is done for you. Here you can see the taxonomy tree of a lab assembled mixture and 1% FDR. So you expect this to be clean and simple because it's lab assembled, but it's actually very complex. If you go to 0.1% FDR, it actually starts to zoom in on exactly the bacteria that are in the sample. So this specificity gain of 10-fold really makes a difference when you try to interpret what's in the data. And the fact that we can now in metaproteomics reach that level is really, really quite impressive, because it will make downstream analysis so much more reliable. It's really quite interesting to see that effect so strongly. How does MS2 rescore do this? Well, because it uses these features that we give it. This is the MS2 PIP features, and here you can see all the different features split out in terms of performance, and the DeepLC features very heavily. In fact, we use them together a lot more than we use the search engine features. So what this percolator learns is that these predictions are incredibly useful to separate between the good hits and the bad hits, and that's exactly what we had in mind. So it's really nice to see here that what we had imagined would be the effect is literally what happens. And this is what gives it the power that it has, actually, the discriminating power. Even more cool, and this is a work that has been done with Christine Carapito and Aurélie Hirschler from University of Strasbourg, who were very happy to provide us, well, they were not happy about this, but they were very kind to provide us with some data. Why were they not happy? Because we forced them to record bad data. So we told them to misconfigure their instrument to set the collision energies too high, which actually messes up the spectra. You can see that here in the explained ion current gets progressively worse, and the Pearson correlations of the Y ions and B ions get progressively worse. And you see the identifications get progressively worse because they're misusing their instrument. Optimal setting is here, and when they go here, it gets worse and worse. However, why did we do this? We do this to see what the effect is on our algorithm, because technically it should learn that these correlations become worse, the spectra become worse. Therefore, it should change the way it scores the data. And you see that effectively happening. The MS2 PIP is the correlation between the predicted and the real spectrum. And as the spectra get worse, you can see that this effect is diminished. So the algorithm has figured out that, wait a minute, these spectra look bad. I should instead use retention time. And you can see that retention time grows in favor of fragmentation prediction. The algorithm, therefore, clearly adapts to the quality of your data. It knows which features are good and which features are not so good, and it adapts. So even if your data is suboptimal, the algorithm will still get the most out of it. And you see the effect here with the gains. The gains get relatively better as the data gets worse, because it adapts to bad data in a very intelligent way. That was a really nice thing to see, actually. Then another thing another thing that I should have pointed out here is that there's not just a gain, there's also a loss. Some identifications are removed compared to the original identifications. And these are very important. Now, these removed identifications, what do they look like? What you can see in green here are the identifications that we have, the total number of good identifications. And in red, you see the decoys. And of course, the decoys don't look like the good hits. That's the whole point of the exercise. This is retention time, which should be at 0. And this is correlation with the spectrum prediction, which should ideally be at 1. And you can see that the good hits are very close to 1. And that here the bad hits, the good hits are very close to 0. That's all fine. The junk is spread out. And here the junk is really at 0 correlation and worse. What you can also see, but barely, is in blue are the guys we kicked out here. And these blue guys, they fall nearly perfectly on top of the decoys. So what we have removed is clearly bad data. And it is because we remove some of this bad data that we can go down to 0.1% FDR. This is the key thing. This is where the specificity increase comes from, because the algorithm can not only say, this is clearly good, it can also say, this thing, which in the past the search engine thought was really nice, actually it's really bad. And it kicks it out, allowing us to go to that 0.1% FDR. Another thing that Christine did was she allowed us to, she made some samples where we could very easily see differences in intensity brackets. So we have high intensity precursor peaks, which the traditional search engines can identify, which Percolator helps with, and which we help with more, but the gains are not that dramatic. And we can go all the way down to the lowest possible signals in the data. And then you see that the low signals are practically not identified by the classical search engine. It really struggles. Percolator helps, but we help more. And so here the gains are much bigger. So what is this telling me? These machine learning approaches are really good at difficult data. Why is that so relevant? Because this is the stuff that everybody has seen. Remember when I showed you in the Campi study that 50%, the top 50%, everybody finds? This is the bottom 50%, and it's the bottom 50% that we have not seen yet. This is the same for immunopeptidomics. This is the same for single cell proteomics. If you want to find things that nobody has found before, they are going to be hiding here. But they will not hide from MS2 rescore. So if you use that tool, you will be able to access this, and you will be the first to access this. It's really quite powerful in that way. So with that, I think I've come to the end of all the stuff I wanted to tell you about. Fundamentally, proteomics has a problem with ambiguity. Therefore, we use databases. When the databases become big, the problem of ambiguity comes back in. Metaproteomics is particularly badly hit by this. And so as a result, the identifications have always been relatively poor. We've shown that the identifications nevertheless are quite robust, especially if you look at the top most abundant proteins for functional analysis. But if you want to dig deeper, you run into some problems, and then the differences become more apparent. And the way to fix this identification problem is to use a fancy tool. And the fancy tool is based on machine learning, and it's called MS2 rescore. MS2 rescore fixes the identification, but makes it better. Makes it better in terms of sensitivity, because you get more identifications, but also specificity, because you get more identifications and you can go to a much lower level of false discovery rate, which actually makes your downstream analysis much more reliable. And then I also mentioned that MS2 rescore does not just work for metaproteomics, it works for immunopeptidomics, it works for single cell proteomics, it works for all kinds of difficult situations. So the final thing I wanted to say is to mention this Metaproteomics initiative that is a part of the European Proteomics Association, of which I am the president. That's why I have to advertise it. And the Metaproteomics initiative brings together a lot of people in the field. There's about 80 or so groups, I think, involved from all over the world. It's not just a European thing, it's really international. And if you're interested in metaproteomics, and if you're interested to engaging with this community, you can just join these guys. It doesn't cost you anything. They do big things, like the Campi study was made by them. And it's a really wonderful way of bringing the community together and working towards solutions and exchanging ideas and best practices. So anybody into metaproteomics, you're very warmly invited and welcomed to contact them. You can see the contact details there, and to just join this initiative and be part of a nice club of people that do amazing things. So with that, I am done. I would be very happy to answer any questions you may have. Just to mention that in the beginning, I showed you the universe and how difficult it is. In Belgium, we have solutions for that. I don't know if you know Tintin, but Hergé drew this rocket to the moon a few years before they actually went. So let's hope that together with all these tools, we can go to the moon. Thank you very much.
Thank you so much for your presentation. Very detailed, very structured. Learned so much. We have some questions in the chat. I would like to ask you to answer them one by one. And for the sake of time, I would like to go to the next presentation. So Leonard, thank you very much. So our next presentation is addressing immunopeptidomics. This presentation will be given by Professor Anthony Purcell, who is talking to us from down under, from the largest Australian university in Melbourne, from Monash University. And I'm happy to to see your contribution today, as I've learned a lot from your contribution to immune response and host pathogen interactions in the past. Looking forward to your presentation.
Okay,
the floor is yours.
Can you hear me okay?
Yes.
Yep, great. Let me just share my screen. Okay, well, thanks everyone for staying around. I know it's probably getting late for everyone. And thanks to the organisers for letting me speak about, I guess, my favourite subject. So I'll try and be brief, and I should thank Leonard in particular for really going through some of the challenges in terms of the computational aspect of what we do. And I've managed to lose a few slides, so hopefully we'll speed things up a little. Today, what I'd like to do is talk about immunopeptidomics, what it is, why we do it, how we do it, and where we're trying to take it, particularly into the clinical context. So I'll give about half background and then show you some data and some new ideas of where we'd like to take the field. There's just a few quick disclaimers about me. So if you think I'm biased, there's probably a good reason for it. So where I thought I'd start the talk is to really talk about the molecules that we study. And these are molecules that are encoded by the major histocompatibility complex, which is a large region of the human genome. It's super variable. It's the most polymorphic region of the genome, and encodes these molecules called human leukocyte antigens, or HLA molecules, and a bunch of molecules with high homology. And back in 1987, I still remember seeing this paper from Pam Bjorkman where they've described the structure of these HLA molecules. So this is the membrane distal region here, and the molecule forms this really nice cargo binding cleft or antigen binding cleft. And I guess for the students in the audience, to give you the idea of the scale of this study, they did 200 litres of cell culture of a particular B cell line. They used papain to cleave these molecules off the cell surface, purified them to get this structure. And the exciting thing about this structure was the visualization of this cleft. So this is now the top view of this molecule. And this density in the antigen binding cleft here for this molecule, HLA2, turned out to be a peptide antigen. This really changed how everyone thought about immune recognition. So I guess if we move on now, understanding all these types of molecules, what transpires is that the immune system is able to sense all aspects of cellular biochemistry. So I know there's a bunch of you that work on lipids, so I thought I'd better throw in something about lipid sensing, others that work on small molecules and metabolites. So what we work on and what I'll focus on today is this aspect here, the CD4, CD8 positive T cells, so the killer cell T cells are the CD8 positive T cells, and the helper T cells are the CD4 positive T cells. And these are able to recognize either MHC1 or MHC2 molecules, or HLA class 1 and HLA class 2 in humans that are presenting peptides in their antigen binding cleft. However, similarly, there are molecules called the CD1 molecules, which are able to present lipids to these T cells and different T cells. There's a molecule called MR1 that's able to present metabolites and small molecules, and in particular vitamin B metabolites to a bunch of cells called mate cells. There's a mucosal associated invariant T cells. And then there's other unconventional T cells that are able to recognize other small molecules such as phosphoantigens in the context of receptors such as the butyrophilins. And the really nice thing from our perspective is that all these types of ligands are perfectly easily studied by mass spectrometry. And in our lab, we're lucky to have several different flavors of mass spec. So to keep the vendors happy, we work a lot with the timsTOF Pro 2, the Explorers 480, and of course, more recently with the Xenotoph. So what do we study in immunopeptidomics? Because we're studying quite a variety of cellular processes, we can study a variety of, I guess, immune scenarios. So everything from immuno-oncology, infectious disease, autoimmunity and allergy. As I've mentioned, we can study peptidic and non-peptidic antigens, restrict to these different types of molecules, and we can work across a whole range of different host organisms. And I've just listed a few there that we've worked on in the past, I guess including the very Australian Tasmanian devils down the bottom there. So this is essentially what we're studying, and in the context of peptide antigens and the HLA class 1 and class 2 molecules, we're interested in studying how cellular metabolism, in particular protein expression, is communicated to the outside of the cell, and in particular how it's communicated to the immune cells. So we'll focus predominantly today on the class 1 pathway. So this is occurring in all the cells in our body that are nucleated. It involves the processing of antigens that are found in the cytoplasm, either because they're endogenously expressed there or because they've come from, for example, a viral infection. They're degraded through the proteasomal system into short peptides, which are then actively transported into the endoplasmic reticulum, where they can assemble with these class 1 molecules, and they can then go out to the surface and present essentially what's a snapshot of what's going on in the cell in the context of the sort of, I guess, the peptide information within the cell that the immune system is able to sample. And of course, when this occurs in our body, we have these CD8 positive cytotoxic T cells or killer T cells. And when a foreign antigen, a peptide derived from a foreign antigen is presented, one hopes that we have a T cell that's able to see these and take out the cell. So I guess to put that in a slightly different context and to sort of think about how this works, I mean, this is essentially how immunosurveillance works. So we have a professional antigen presenting cell, in this case, a dendritic cell. It finds an antigen, it eats it, it breaks down that antigen into peptides, and then it shows off that antigen to T cells to then go off and find the infected cells or the cancerous cells and kill them. The other pathway that occurs in these professional antigen presenting cells is the class 2 pathway. This is much more specific to exogenous antigens, and most of the professional APC have some sort of phagocytic activity. So they're constantly sampling antigen in the environment. And these peptides are degraded in the endolysosomal compartment. They're able to bind to nascent class 2 molecules, go out to the cell surface. And in this case, they're recognized by these CD4 positive or helper T cells. And these T cells are both responsible for licensing immunity, so for sort of really activating all the immune cell subsets, and also for antibody production. So when we study this really complex mixture of peptides bound to HLA molecules, we call this the immunopeptidome. And I'm sure we're all familiar with that now. And really this is just presenting a snapshot of the cellular proteome. It's influenced by the presence of pathogens and malignancy, which is why we're interested in studying it, because we're interested in both infectious immunity and cancer. And today, most of the examples I'll show you are in the immuno-oncology space. The composition of these peptides is highly dependent on the background genetics of the individual. So we all will have different HLA molecules that we express. We may share 1 or 2 allotypes, but we have 6 different class 1 and 6 different class 2 allotypes that we express in our cells. So there's a very distinct chance that no one listening to this is going to select quite the same peptides for presentation. And this really is to highlight the basis for why some people are more susceptible to viral infection than others, depending on their MHC haplotype or the HLA haplotype, and the sorts of viral peptides, for example, that they can present. So it's highly personalized and therefore complicated to study. It's also influenced by the cellular microenvironment, and things like inflammation are able to remold the sorts of peptides that are presented through modulation primarily of proteolysis within the cell, the induction of the immunoproteasome, which is an inducible form of the proteasome, and also with upregulation of some components of the antigen processing machinery. In particular, in recent years, we've been very interested in the dark immunopeptidome. This presents its own challenges, not only because some of the components are quite controversial still, but it's also extending, I guess, the sort of database we need to search. And Leonard's already discussed this. If we start to consider a lot of the non-canonical sources of antigens, such as endogenous retroviral elements, novel open reading frames that aren't necessarily contained within the standard sort of databases. So these are things like gene fusions, regions that are from non-coding RNA, which actually do code for peptides and proteins, and other sorts of different reading frames. Post-translationally modified peptides adds additional complexity to what we might want to study, but a number of these PTMs can be quite specific to cancer cells in particular. And this can include the much maligned proteasome catalyzed splice peptides, which I'll speak very briefly about. But also when we're working with clinical samples, we need to consider environmental and microbiome-derived peptide antigens. So I guess we're doing immunometapeptidomics, Leonard, for want of a better term. So this is just to reiterate the fact that your HLA haplotype will dictate the types of peptides that are selected. And this is because the vast majority of polymorphism occurs within this antigen binding cleft, which impacts on the sorts of peptides that are selected. And I won't dwell on this too much, but the different alleles we have different shaped and different charged pockets because of these polymorphisms. And this selects for different types of peptides. So here, for example, we're able to see that for HLA-A1, we often see an acidic residue at position 3 and a tyrosine at position 9. A similar molecule with different polymorphisms, HLA-A2, one of the most common HLA molecules. Here we see a leucine or methionine at position 2 and a valine or leucine typically at position 9 of the bound peptide. So depending on which allele you might inherit from your parents, you will select different peptides and therefore different parts of different pathogens or different tumor-associated antigens. And this obviously presents challenges when we're trying to think about how do we make broadly applicable antigen-specific therapies when everyone's going to select different peptides and therefore would need different peptides to stimulate appropriate immune responses in those individuals. As I've mentioned, these are encoded by the major histocompatibility complex, and this is this region that contains all of these molecules plus a number of other immune-related genes. So I thought I'd talk a little bit about the sort of diversity of antigens, and I've already alluded to some of these already. And I thought I'd just quickly run through them because this really highlights the sort of complexity we want to visualize within the immunopeptidome. So we have the sort of constitutives. So these, I guess, are the database encoded antigens, proteins that are expressed from the genome, polyubiquitinated, typically degraded by the proteasome into peptides that can be selected by the various HLA molecules we express. In the context of cancer, we might have specific cancer related mutations. So we need to sort of have a more bespoke database or a sort of proteogenomics approach, if you like, to be able to identify these types of peptides that are degraded through a very similar mechanism. The various cryptic sources of antigen, these non-canonical sources of antigen that I've briefly alluded to, including so-called DRIPs or defective ribosome initiation products. So this is where there's some sort of aberration during translation, and these short peptides are produced during an aborted translation event. Often there can also be introduction of a an inappropriate amino acid. So you're sort of making a sort of random, if you like, peptide. And then the so-called PTM antigens, so things like we know that phosphorylation, for example, alters the way that proteins are degraded by the proteasome. So you can liberate different peptides depending on the post-translational state of the molecule. And then these peptides that I'll touch on very briefly that we believe are produced within the proteasome, where instead of getting hydrolysis of a bond during degradation, there's the potential to actually get a reverse proteolysis reaction or a ligation of two distinct peptides that aren't linearly encoded in the genome. We know that PTMs are typically quite abundant in the immunopeptidome, and this is just some relatively old data, just showing that different alleles have different proportions of different types of PTMs, and I'm sure you'll see many of your favorite PTMs within this list here. And what we know is that the PTMs can influence both binding to the HLA molecule, but also can be solvent accessible and accessible to the T cell receptor on T cells, and therefore influence how the T cells perceive that particular peptide antigen complex. We're also very interested in the sort of heterogeneity that's introduced in this system. So we work in a system where there's no fixed digestion step, if you like. These are naturally produced peptides, and there's heterogeneity in their production because there's cleavage in the proteasome, there's various peptidase trimming in the endoplasmic reticulum in particular, and this can lead to peptides of varied length from the same, I guess, starting antigen. So for example, when there's a lack of precision in trimming, we often see when we purify these peptides, not just a sort of core peptide sequence, if you like, but also we often see N or C terminal extensions of these peptides. And this is due to this heterogeneity. So again, there's this complexity in how we need to measure these things, and obviously then complexity in the database search, where we have to basically consider a very large search space. And we know when there's this length heterogeneity, two things can happen. So I guess quite traditionally, it's been thought that as the peptide length increases, the peptides sort of constrain within this antigen binding cleft, and therefore, as the peptide length increases, the peptide starts to bulge out. And we've seen very nice evidence through structural biology of peptides up to 16 amino acids in length in the class 1 molecule, producing really quite pronounced bulge structures, which obviously present then a challenge for the T cell receptor to accommodate as it's docking on the top of that HLA molecule. And then more recently, we and others have shown that the peptide can also overhang the cleft of the class 1 molecule. And this sort of went against, I guess, the paradigm that this is a closed cleft and that the peptide sat in the cleft like a sausage in a hot dog roll, if you like, that was able to potentially hang out as well. And in some sort of recent data that we've generated, we've shown at least for a HIV epitope that length does matter, and that in fact when we look at the sort of abundance of these peptides, and this is, I guess, just looking at intensity, we haven't done any absolute quantitation here, that the minimal peptide epitope, this TW10 epitope here, is probably less abundant than some of its more extended forms. And when we look at different T cell clones from HIV-infected individuals, we're able to show that for some clones, at least, they actually are quite sensitive to the length of the peptide, and actually prefer to see some of these longer isoforms, suggesting there's a functional consequence of this heterogeneity of length of peptides that are presented. So that's, I guess, a bit of rationale around what are the sorts of molecules we want to see, but how do we actually go about identifying them? And so we've published some of this methodology quite extensively. But I thought it'd be good just to go through this and sort of give you a feel for the experimental workflow, talk about some of the challenges, and then show you some, I guess, some specific examples. So this is a general method when we have a reasonable amount of material. So typically, if we're working with cell lines that we can grow in culture, or if we're working with reasonable amounts of tissue, so let's say 100 to 500 milligrams of tissue, that could be a resected tumor. It could be an organ or tissue from either an infected individual, or from a healthy individual, for example. So the whole process starts off with very gentle lysis of the cell, and we try and lyse the cells and keep these HLA peptide complexes in their native conformation. We then apply this lysate to an immunoaffinity column, and this column typically consists of some sort of bead. We like to use proteinase separase because we get regio-specific capture of our antibody. The antibody is then cross-linked to the resin, and we then pass our lysate through. And we're lucky that we have a good range of different antibodies that are able to capture the different HLA complexes that are presented on the cell. So we might use an antibody such as W632, which is predominantly used in the field, that captures all class 1 complexes, for example. Or we can use specific antibodies that capture different HLA allotypes. So for example, there's an antibody called BB7.2 that captures specifically the HLA-A2 complexes from these lysates. Once we pass the lysate through this first column, we're able to have columns in series to capture other types of complexes. So we might capture A2 followed by a pan class 1, followed by columns that will capture the class 2 complexes from the cell surface if it's a professional APC. We can then decouple those columns and then proceed to elute the peptides off the column. So from our affinity matrix here, we've got our captured HLA peptide complex, and we treat this with an acid solution that obviously sort of elutes the HLA peptide complex from the immobilized antibody, but it also leads to association of the HLA protein part and the peptide cargo. And we're obviously interested in capturing that peptide cargo. So typically when we have a large amount of material, we do some sort of reverse phase capture. We particularly like to do this on a sort of microproperative HPLC, because we're able to see the peptides coming off, not particularly impressively, but we can see them just looking at the 215 nanometer detector. But we can also see the heavy chain, etc., come off as well. And for class 1, we see a beta-2 microglobulin peak. This is the monomorphic light chain, which allows us to quantitate the amount of material we've actually captured in this step, and it's a good quality control step in this process. We then take individual fractions and analyze them by mass spectrometry. We do our database search to identify the peptides, and this is a very peptide-centric workflow. I'll talk about that in a moment. And from that, we can also infer the source antigens, and we then like to analyze sort of HLA peptide characteristics and validate them. So typically, because we know the immunogenetics or the HLA typing of these cells, we're able to partition peptides into their various allotype-specific binding motifs. Also look at length distribution to get a good feel for the quality of the peptide identifications. And I'll show you some specific examples a little later where you can see how that's done. But there are key challenges, and Lynette's already touched on some of these. So firstly, these are naturally generated peptides. So there's no defined enzyme specificity. So suddenly we get this huge increase in the search space, even looking at a standard sort of reference proteome. Not all the algorithms we use cope very well with the no enzyme workflow. And typically we have to restrict our post-translational modification search in the first screen, because if we add PTMs onto this no enzyme specificity, the search space becomes almost untenable. Often only 1 to 2 peptides are found per protein in the immunopeptidomes. This is very different to the proteomics workflows that you're used to, where you tend to accumulate evidence for a protein and it gives you confidence in the peptide-based identification as well. Here, everything is peptide-centric, and this requires high spectral quality, which means we have to tweak both the chromatography to get good resolution and very sharp peaks, but also we have to tweak the MS acquisition and probably spend a little longer fragmenting and acquiring fragment ions than you may for a tryptic peptide, for example. And this is just, I reiterate here, the peptides from a given allotype are very similar length. Predominantly for class 1 peptides, they're 9 Ms. And they share these common biophysical characteristics because they have these shared residues that are involved in binding to the cleft. So particularly for HLA molecules at position 2 and at the C terminus. And our sample preparation typically takes about 3 to 4 days. It's not a quick process because of the multiple capture steps and extra fractionation. So this is the strategy we use particularly for our cancer epitope identification. To sort of give you a feel for the sort of workflow. So we will take our tumor tissue or patient-derived cell lines. For the cell line work in particular, we'll often treat with different cytokines because this really expands the number of peptides we'll identify because of the plasticity of the peptides presented and changes in sort of the proteolytic activity of the proteasome in particular. We'll combine some sort of genomic analysis with our LC-MS data to get a sort of tumor-specific database. And then in particular, we like to use peaks, although we use other algorithms as well. MS-Frago, for example, works very well to sort of interrogate our data. We really do I guess, take advantage of the de novo assisted aspect of the peaks algorithm, in that it sort of pays more attention to the MS spectra and potential candidates than necessarily doing peptide spectral matches per se. And with the de novo sequencing, we're able to identify neoepitopes, even if we don't detect the mutation here or we don't have genome sequencing. And often when you get a sample from a collaborator, you don't get any genomics at all. We're able to identify non-canonical antigens that maybe aren't present in our database. And we can also identify these spliced peptide antigens that are sort of impossible to predict and really essentially random sequences of pieces of linear sequence stitched together. We also take advantage of a sort of secondary search where we open up the types of PTMs we might be interested in to look for cancer-specific PTMs within this data set as well. So if we do that, what sort of numbers can you expect? And this is a question I get asked a lot, so I thought I'd just put some numbers out there. So if we go really deep with a patient-derived cell line with multiple treatments, typically we anticipate we will see somewhere between 50 and 100,000 unique peptides per cell line at a 1% false discovery rate. If we work with tissues and biopsies, this number drops significantly by about 5-fold. And I've just sort of listed here, I guess, some of the things that we're thinking of. There were a few slides inserted here, but Leonard did such a good job of talking about the way we handle data. I thought I'd just restrict it to talking about we are using these large data sets to generate spectral libraries that are allotype specific, so we can start to do data independent acquisition. We also are very interested in AI-based approaches to improve our PSMs and particularly for these peptide-centric workflows. So incorporating things like retention time prediction, MSMS, so predictors of MSMS spectral intensities of the fragment ions. When we're using the timsTOF, sort of really utilizing all the other properties we can measure, so things like CCS, and also incorporating various de novo sequencing algorithms, including the one in peaks. And what this allows us to do is really, I guess, have a totally source agnostic view of the data we generate. So within the tumor antigens, we can see the cancer testis antigens, the neoepitopes, as well as things like post-translational modifications. And these can all be quite tumor specific when we're able to compare tumor tissue, for example, to healthy adjacent tissue. So I thought I'd better show a little bit of data. So this is just this is a vignette of the sort of data that you can generate. And then this is a project that a couple of master's students did a few years ago, and it's just hopefully about to be published, where we've been comparing colon cancer ligandomes. And in particular, the goal of this study was to compare microsatellite stable versus instable phenotypes. So the microsatellite instable phenotype has a DNA repair defect, and therefore these tumors accumulate a much higher burden of mutations. And with the high mutational burden, this leads to improved patient outcomes because there are more targets for tumor infiltrating lymphocytes to recognize, and therefore greater responsiveness to checkpoint inhibition. I guess what we really wanted to do is sort of really confirm at the immunopeptidome level that this is indeed the case. So we compared 2 cell lines from the microsatellite stable phenotype, and these 2 patient-derived cell lines are derived from the same patient, one from the primary tumor, one from a lymph node metastasis about 12 months later. And when we looked at the peptides we could detect, we found around 32,000 peptides across these cell lines. This is just showing the overlap. And remember, these are from the same individual, so we'd anticipate there'd be a good overlap. But what you can immediately see with the metastatic cancer is a reduction in the total number of peptides, because the metastasis is essentially just down-regulated a lot of its antigen presentation machinery. When we look at these peptides that are predominantly 9-mers, we're able to extract out some of the key peptide binding motifs from the HLA molecules expressed by this tumor. So we can identify the peptides that are bound to HLA-A2, for example, based on this sort of motif of a leucine methionine at position 2, failing leucine at the C-terminus. Similarly for some of the other HLA molecules, which have quite characteristic binding characteristics as well, and quite distinct from each other. What we do find, though, when we look for mutations, and we identified around 1,500 potential peptides derived from mutations from these cell lines that potentially could bind one of these HLA molecules, we could only really find one mutated peptide out of the 32,000 we identified, which was a bit of a poor return on investment. But when we consider other types of peptides, in particular post-translationally modified peptides, we found that this was actually quite a rich potential source of peptides from these particular cancers. And importantly, around 2,000 of these post-translationally modified peptides appeared to be cancer specific, in that they were derived from cancer associated or cancer specific antigens. So whilst there aren't many targets for neoepitope specific T cells, there is potential to think about bolstering immunity to some of these post-translationally modified peptides. If we then move to the MSI, so the microsatellite instable phenotype, and this is data from 2 cell lines from unrelated individuals, although they do share the HLA-A2 allele. This is important as the story develops here. Again, what you can see when we use either BB7.2 to pull down the A2 specific peptides, and that's what we're going to focus on for the next few slides, or for the rest of the class 1 molecules using this pan class 1 reagent, is that we see predominantly 9-mers are bound to these molecules, which is what we anticipate. And we can extract out, for example, with the BB7.2 pull down, a nice HLA-A2 motif for all the bound peptides. These are the sorts of numbers we get. Obviously, HCA7 was a bit more generous in terms of the number of peptides, and there's a significant overlap in the peptides we detect, which really represents the overlap in the HLA-A2 peptides that are presented by both cancer cell lines. I guess importantly, we saw this expected increase in the number of mutated peptides we could identify. So to remind you, in the microsatellite stable cell line, SW480, we could only find 1 mutated peptide despite there being multiple. We're able to detect considerably more in these 2 cell lines. And I think really excitingly, for the A2 peptides, we found 4 shared mutated peptides. And these were predominantly in this type of cancer. These are predominantly frame shift mutations where, and this is related to this defect in DNA repair. So, you know, this is kind of one of the holy grails, if you like, of trying to identify these cancer-specific neoepitopes to try and find those that are shared between multiple cancers and individuals having, I guess, a common allele, such as HLA-A2, which is found in around about 40% of the Caucasian population at least. So that's, I guess, an example of the sort of data you can generate. What I thought I'd do in the last part of the talk is talk about where we're trying to take this. And, you know, it's all very well to work with cell lines, patient-derived cell lines is a little better. Working with large amounts of resected tumor, for example, is also possible. But often that's not what happens clinically. And so what we wanted to do was really, I guess, take our workflow and try and make it as relevant to the sorts of clinical samples that we're being asked to analyze. And that typically is, in best case scenario, a core biopsy, so roughly around 20 milligrams of material. And often with these biopsies, the proportion of tumor is quite low. So we want to get the best sensitivity we can. So we've taken our workflow and miniaturized it. And this in particular is through the use of these Mobicol spin columns, where we're using around about 10 microliters of resin with our various antibodies. This can also be done in series, so we can look at multiple specificities in the pull down of the HLA complexes. We use acid to elute these from these small columns in very small volumes, and we use these Northway cutoff filters to separate the proteinaceous components from the peptides. And because we wanted to go to better and better detection, we decided to sort of, I guess, take some inspiration from the work in single cell proteomics, where peptide barcoding using things like TMT labeling and adding in a sort of carrier peptidome would allow us to amplify the signal. And at least when I explain this to the clinicians, we call this sort of a sort of peptide PCR, in that we're able to use an abundant source of the peptide to amplify the signal from these very small amounts of clinical material we're analyzing. So I probably don't need to explain this to this audience, but for example, with the TMT labeling, we're able to combine samples, which gives us an additive signal in our MS1. And if we add in a carrier peptidome, so this can be from various sources, and I'll show you some examples of this, where an abundant source of the HLA peptides, we can really get significant improvements in the MS1 intensity. And of course, when we do our fragmentation, This gives us additive signal in the structurally informative part of the MSMS spectrum, but also allowing us to get information about the abundance of the various contributors to this MS1 peak. So typically we see our two carriers here giving us very nice signal, but also with some of the other tags giving us small amounts of signal, suggesting that for these particular samples, there was a very small amount of this peptide present that probably wouldn't have been sufficient to trigger an MS2 event, except in the context of having this carrier boosted triggering of MSMS. And this can be used both in terms of yes, no, sort of yes, we can detect the peptide in this sample or not, but also to an extent gives us an ability to get quantitation as well based on the TMT channel intensities. So what does this look like? So we sort of did, I guess, a proof of concept experiment where we took our favorite B cell and so titrated in the input from 5 million cells down to 1,000 cells. To give you context, I guess typically we work with around about 5 times 10 to the 7, so 50 million cells or more to sort of do our discovery-based workflow. And I guess I wouldn't be telling you this if it didn't work. So this is the immunopeptidome from 1,000 cells using this barcoding approach, and obviously using the more abundant source of the same material to amplify the signal of the peptides in the 1,000 cells. This is just an example of a B7 peptide, B7 peptide from this, and you see we get reasonably good linearities as we start to go down. And nicely here we did a dual pull down of class 1 followed by a class 2 column. We're able to identify the class 1 peptides with a typical length distribution dominated by 9 Ms, and the class 2 peptides, which are typically longer with a sort of median length of around 15 amino acids. And we can extract out nicely some of the binding characteristics of the different peptides here. So at 1% FDR, we're visualizing around 900 peptides. And I guess from 1,000 cells, this really is a very small amount of input. So what does this look like? I guess in a slightly more, but still contrived clinical situation. So we're sort of fortunate to have a very strong oncology department at Monash, in particular working with Mark Shackleton, who spends a lot of time making patient-derived xenografts and expanding these very small parts of a biopsy in a nude mouse. We were able to access both a mouse PDX grown from a biopsy, but also the remaining amount of the biopsy. So this was a core biopsy, and there was around 20 milligrams of this biopsy left. So what we decided to do was to titrate in the amount of the PDX and use this as the carrier in the carrier channel, and then take the biopsy and take 19 milligrams of it, label it with 1 TMT label, and then take a very small amount of the biopsy, around 1 milligram, label it with a different TMT label, and search our data and ask, well, can we detect peptides that are of any clinical utility from that 1 milligram of biopsy material, or the 19 milligram, which was our sort of backup position. And again, just from the 1 milligram of biopsy material, we're able to identify around 900 class 1 bound peptides. And again, these peptides exhibit classical length distribution for class 1 peptides, but I guess more importantly, we were able to sort of get some insight into the sorts of potential targets that are being expressed on this particular melanoma. So here you can see the quantitative information in the heat map here for a number of cancer-specific peptides derived from these various fairly well-known T-cell targets. So all these peptides are able to induce T-cells in various settings, suggesting that this particular melanoma had potential to be quite immunogenic and present peptides from a number of known tumor antigens. So that's all very well, but unlikely that you're very often going to have a nice PDX to act as a carrier for these types of samples. So we decided to sort of think a bit more in terms of clinical context, and we took a number of primary dissociated cancer cells, adenocarcinomas from endometrial, colorectal, ovarian, and lung cancer. We isolated the A2-bound peptides and labeled them, and then we took a carrier that consisted of a number of peptide antigens that we were interested in. So these are known HLA-A2 restricted T-cell determinants. And just to sort of, I guess, very quickly show you what that looks like. So we had 187 peptides of interest. So these were all known T-cell epitopes. We're able to very quickly, with these various cancer samples, screen for the presence and abundance of these particular peptides, and we were able to find evidence for 129 of the 187 peptides in at least one of the eight primary dissociated cancer cell samples. And these dissociated cancer cell samples were around about 1 million cells in total, which is about what you'd anticipate to get from a typical biopsy of a cancer. So hopefully I've shown you that some of the new workflows, we can use significantly smaller numbers of cells, and this has always been a challenge in the immunopeptidomics field. I've shown you that at least in MSI high colorectal cancer, we can see substantive numbers of neoepitopes. And these are often very difficult to find in the immunopeptidome because they typically are less abundant than everything else that's presented. I've shown you that some HLA-A2 restricted neoepitopes can be shared across unrelated tumours, which I think is really encouraging for those that are interested in developing targeted immunotherapies that work in more than one individual. So these medicines that are more broadly applicable. Potentially splice peptides as a novel target for microsatellite stable colorectal cancer. I've sort of introduced this peptide PCR concept and its potential utility in the clinical space, particularly where you may have off-the-shelf immunotherapies that are targeting known tumor antigens, and a very, I guess, reassuring thing to do would be to make sure that the patient biopsy actually is presenting the peptide you're trying to target. And yeah, I think sort of excitingly from my perspective is that we really are starting to see the emergence of clinical immunopeptidomics and its potential to translate into truly personalized approaches for cancer immunotherapy and other types of immunity as well. So it just remains to thank the people involved. I won't go through all their names. There's a lot of them. To thank our funders and to thank you guys for hanging around.
Thank you very much. Very nice presentation, a lot of new data. If I am allowed to ask a first question, I'm fascinated by the genomic diversity of cancers. The research on cancer mutations showed that there is an accumulated number of mutations with a different genetic penetrance. So it means we see the mutation, we don't know where is it showing up. And actually, I was puzzled by your presentation that in some of your models, you have not detected epitopes which are coming out of the mutated proteins, which are in thousands. This is a number in breast cancer and prostate cancer samples. But they are rather small, but you have seen a lot of differences in the cancer-associated peptides. So there were differences, but they were not related to mutations. Is it a problem of detecting the peptides, or is it a problem how we understand the genomic penetrance? What do you think?
Look, it's a good question. I think it's probably a combination of. So certainly with these mutated peptides, that they tend to be lower abundance than other sources of the peptides. But what is typically been seen by the field, both in terms of not only the the immunopeptidomics, but also sort of, sort of big efforts to sort of map and in trying to induce T cells specific to these peptides is that a lot of these peptides aren't generated. So you can't sort of linearly translate from the mutated genome to the proteins that are produced, and then from the mutated proteins to the peptides that are liberated, because I think these are very unstable, and potentially, even though there's a potential epitope there that has the right binding characteristics containing that mutation, for whatever reason, they don't survive antigen processing and presentation. So maybe they get chopped in half or something else. So typically, I think the depressing numbers are that probably about 1% of the predicted mutational landscape is translated into what's presented on the cell surface for immune recognition. And we're probably detecting a portion of that. I think, with some of these newer targeted approaches, if I had an unlimited budget, for example, I'd probably make every single peptide TMT label it and go in as sensitively as we can and look. Unfortunately, I don't have that budget, but we try and do our best.
Hi, Tony. Thanks a lot. I have a related question.
Yeah.
In your biopsy analysis, I think it's very interesting. But the biopsy has different cell types. The tumor cells and non-tumor cells. Do you worry about those peptides from non-tumor cells?
Not really. I think if we're interested in asking the question, is this tumor expressing KRAS peptide, for example, or whatever? If there's additional peptides contributed from the healthy tissue that isn't tumor, that probably helps us in some respects because it acts as a sort of buffer for sample loss. What we can't address easily, and I think it's still going to be a major challenge, is when we have tumor heterogeneity, where we know that some of the tumor might be presenting an epitope, others not. And this is the real challenge, and this is what we're seeing in some clinical scenarios where you give a targeted immunotherapy, you take out 2/3 of the tumor, and what grows back is something that's not expressing that antigen. So that's the big challenge. I mean, how many targets do you have to have so the tumor can't escape that sort of immune attack? And so this is why I think most clinical trials now are combining everything with checkpoint inhibition, and you hope that if you get enough tumor cell death, that will be immunogenic and stimulate a broader immune response to the rest of the tumor. But look, I think if there, as long as there's some tumor in there, I think we're okay. And I think yes, there's all sorts of stuff in these biopsies. And depending who's taken it and where it's from, some stuff that's not cellular, some stuff that you can't solubilize at all, and some stuff you don't even want to think about, I'm sure.
Yeah, maybe you can integrate with single cell transcriptomic data or genomics data and integrate it of your mass spec data to get some insight.
Yeah, I guess because we're looking at a global cellular population, probably just global RNA-seq, whole genome sequencing is fine. The single cell data, I guess, is very nice, but it's probably not really going to address our issue because we're not doing immunopeptidomics on that single cell.
You can handle 1,000 cells, which are very amazing. Actually, I have a question. In addition to the cells, can you detect them in fluids like plasma or urine? And can you detect them in FFP tissues? FFP has a formalin fixation. I don't know whether it's possible to analyze those because there are a lot of tissues that are in FFP format slide section.
So I'll try and address the two questions really. So in terms of body fluids, you can detect both soluble HLA, which is shed from the cell surface through metallopeptidase activity. You can also, it's also quite abundant in exosomes. which you can find in the body fluids as well. So you can definitely do like, I guess, a liquid biopsy, if you like. And there's certainly some evidence, not so much from us, but others that has shown that you can detect some cancer-specific antigens, some viral antigens being presented on this soluble HLA. So I think that's quite promising in terms of thinking about, you know, if it was urine, for example, you can collect quite a lot of that easily and non-invasively. In terms of formalin fixed tissue, I mean, that's always a challenge for everyone. I think the only way I can see it working and what we're trying at the moment is a very targeted approach where we're not going to be able to capture the class 1 from that tissue, but we can probably acid dissociate enough peptide for detection. And at least our first few efforts, I mean, you find huge amounts of oxidation, weird cross-links, all sorts of stuff on the peptides as well. So I think that's still going to be the ultimate challenge, and it'd be nice to be able to work with that material, but I think the information content is going to be restricted to probably a really focused, targeted assay for, you know, a handful of peptides of interest.
So have you analyzed the secreted medium of cell culture? You know, if they are shedding, probably can even detect. With body fluid, probably too complicated, but cell line probably can be a good benchmark.
So we've done plasma, we've done urine.
Yeah, plasma urine may contain shedding peptides from all the organs. But if you do a cell culture, and the culture media may only contain the peptides from the cancer cell that you're culturing.
Yep. So look, if you use serum-free media, you can certainly detect both exosomal and shed material. And if you treat your cells really mean and they undergo apoptosis, they tend to shed a lot more HLA. So, you know, different people get different yields, and it's not always a good thing. But you know, look, it's all certainly possible.
That's the idea. So for the PTM analysis, you mentioned a few PTM, but have you exhaustively analyzed all the possibilities? There are a lot of potential PTMs.
Well, I think, yeah, so I think the Peak software allows you to look for up to 500 different PTMs. Some of them, I don't even understand what they are. Super weird. To be honest, I mean, when we've looked, the ones we detect reliably and believably and that we've validated are things like deamidation, phosphorylation, acetylation, methylation, etc. We haven't sort of, I guess, exhaustively looked by any extent. We've been very interested in, but I was interested in the talk on redox proteomics because we're quite interested in cystine modifications, and these can quite often be related to sort of inflammation. So things like S-nitrosylation, glutathionylation, etc., cystanylation, all these things you can detect. But yeah, I think when you do that, you sort of have to do this two-stage search where you search very specifically for a handful of modifications so you can get quite good specificity. If you try and look for 200 plus PTMs in the raw data, I mean, you're really going to struggle. So we tend to do a database search using a reference proteome, take out all the easy peptides, if you like, or the easy spectra, and then focus in on the spectra that look like they're good quality in terms of signal to noise, and then use these more extensive PTM searches. But you know, it's still very bespoke, and I'd love a tool that you could just put the data and press a button and it might spit out. And maybe Leonard's got one. I was very impressed with his talk and some of the tools he's developing, because the trouble they have with proteomics is exactly what we have with immunopeptidomics. And you know, I think there's sort of recently been a sort of merging of some of the thought processes, and hopefully at the end of the day we'll end up with some really nice tools that we can gain better confidence in our peptide IDs. You know, for us at least, for the clinical work, we have to go through this exhaustive process of making isotope labeled peptides, spiking them in, showing they co-elute, doing all that sort of validation. And at some point, you either run out of budget or you just run out of patience or instrument time, because if you're identifying hundreds and hundreds of interesting peptides, some of these isotope peptides are $400 each Australian. I mean, whoops. And it just becomes prohibitive. So I think tools that give us greater confidence, and I guess as confidence develops, hopefully we'll be in a position where we can at least restrict a peptide synthesis to the really good clinical candidates, and not just to convince us that what we're doing is real, if you like.
Thank you. Are there any other questions from the panelists or from the audience? Thanks so much, Tony. Very nice talk.
Thanks, everyone.
Tutorial. I did a lot. Thanks. Jersey.
So I think we're closing right now the session. I would like to ask Tusi to say the official words.
Thanks, Eric. No, thank you so much for all the presenters today. And it's wonderful to see so many different aspects, and especially the staying after what we plan and hanging and asking questions. Thank you so much, and looking forward to see you all tomorrow. Hopefully we can make the time properly. Yeah. Thank you, Tony and Leonard. Yeah. Hope to see you all.
Thank you. So may I ask organizers to stay online? Organizers, please.